Sunita Singh, Chemistry Department, welcome you all on the second day of International Conference on Chemistry for Health, Hygiene and Environment. Before starting our today's session, following our Indian custom, first we offer our prayers to the Goddess of Knowledge, Saraswati Ji, with a Saraswati Vanna by Tanu Kashyap, student BSc 4th semester. Thank you, Tanu. Now we will start our second day first session with dedication of our keynote speaker, Dr. Sanjay Kumar. I would like to call upon Dr. Navyuga for his welcome and kind introduction.
A warm namaste to all present over here. It is amazing and wonderful that so many people from different time zones have gathered to make some deliberations on some very pertinent questions from the area of chemistry. And the most interesting deliberations have been related to finding new medicines. So here I feel honored to welcome our keynote speaker, Dr. Sanjay Kumar Pandey from the world of pharmacology. He has a very illustrious profile, and may I introduce you to his illustrious profile. Dr. Sanjay Kumar Pandey is the founder and chief scientific officer of Triangulum Biopharma, San Diego, California, USA. He leads the scientific effort at Triangulum Biopharma in the area of NASH, that is non-alcoholic steatohepatitis, diabetes, obesity, muscular dystrophy, and several other therapeutic areas. Triangulum Biopharma serves over a dozen companies and organizations in USA, including NASA, Pandey, Biolorus, and Ionis Pharmaceuticals. Triangulum's scientific research has helped many small companies be acquired by larger pharmaceutical companies. Dr. Pandey is a prolific researcher with 30 years of research experience spanning metabolism, oncology, and muscle diseases. He has co-authored over 25 peer-reviewed publications, including Nature, Cell Metabolism, and Science Translational Medicine, 70 abstracts, and several books and chapters. As per the Google Scholar Annual Top 10 Research Papers in the past 10 years, between 2006 and 16, one of Dr. Pandey's paper is a classic top-cited paper ranking second globally in the field of molecular biology. In addition, Dr. Pandey has been a reviewer for several journals and has been listed as an inventor on many patents. Previously, Dr. Pandey led diverse projects in various therapeutic areas at Ionis Pharmaceuticals, California, and during his 14-plus year tenure, he developed notable collaborations with academic institutions, including the University of Rochester, the University of Maryland, the University of California, San Francisco, the University of Florida, University of Laval, Massachusetts General Hospital, Harvard Medical School. He also collaborated with biopharmaceutical companies such as Eli Lilly, Amgen, Antipogen Australia, Sanofi, Genine, AstraZeneca, and Biogen. Dr. Pandey's preclinical research led to the identification of several compounds that advanced to clinical trials. He holds a PhD in physiology, biochemistry focus from Faculty of Medicine at University of Montreal, Canada, MSc degree in biochemistry from University of Ottawa, Canada, and a BSc honors with chemistry from BHU, Banaras Hindu University, India. Following his PhD, Dr. Pandey was a Fugate International Fellow at National Institute on Aging, a division of National Institute of Health in Maryland. Just before this, I was looking into the abstract that he is about to talk about, the paper that he's going to talk about. So today he's going to speak on the previously undruggable areas, previously undruggable targets, which were unthought of, that world would be able to bring some relief to these undruggable targets. He's going to talk about them. And so I would request Dr. Sanjay Kumar Pandey to take it forward from here. Thank you so much.
Mute her, sir.
Sorry, thank you. So thank you, Aruಣಿmaji, for a kind introduction. Now, everybody can hear me? Okay.
Yes, sir.
Okay. So, namaste everyone, dear faculty, students, and guests of Navyuga Kanya Mahavidyalaya. It's my honor today to join you all in this conference. I feel privileged to be amongst prominent scientists as well as upcoming budding scientists. It has always been my desire to promote and foster science at an early stage. Navyuga Kanya Mahavidyalaya is a great example of the empowerment of women and what can be accomplished with a great education in hand. My father was a professor of medicinal chemistry in Department of Pharmacy, IIT BHU, who after retirement joined as a head VVS Purvanchal University, KN Modi College in Meerut, and lately till he passed away in Saroj Institute of Management as a Dean of Pharmacy in Lucknow. Some of you may be knowing that. And he too had a similar desire. He instilled in us the love of science. He wrote several books in the field of chemistry, and at the end of my talk, I will show one slide where you can see his books, and also I will, share with you. The theme of the conference is timely one, International Conference on Chemistry for Health, Hygiene, and Environment. There are basically 3 to 4 drug discovery platforms, small molecules, antibody, and nucleic acid based, in which scientists design molecules to target disease-causing entities. It could be either protein or RNA or DNA. The drug design is key aspect of any drug development. I hope with this conference, you all will gain valuable experience and apply in your scientific life. I would like to thank Vandana Dwivedi ji, who initially contacted me to make a presentation and introduced me to Dr. Neha Agrawal ji. Dr. Neha Agrawal ji and her team has put together a well thought out program for the conference and deserve much gratitude. Thank you. So with that, I would like to share my screen. Hold on a second, just give me a second. I think my... So I... Let's see. Oh, I think there is some.
Sir, it was visible. Sir, your screen is visible.
Screen visible? Can you? Okay.
Yes, sir.
It is visible.
Okay. I want to make it bigger actually. Can you see it now?
Yes, sir, it's visible.
But I can't see anybody. Anyway, I will try to see if I can. Hold on a second. Is it complete visible?
Yes, sir.
Okay. So let me. So this is the topic for today's presentation is RNA-based therapies and emerging platform. So the agenda for today's presentation is an introduction, and the basics of biology is important. And then the third bullet has therapeutic platforms. I would focus on antisense oligonucleotide. And then I have a very nice example about personalized medicine. So what do we see here? A medicine in a capsule, the ultimate cure. So chemistry plays such an important role in making drugs which could be, put into the tablet or in the form of a capsule. So now in this slide, what I'm showing is the most expensive drugs of 2019. So on the left side, left panel, you will see all the disease indications. And on this side, you see the name of those drugs. The first drug is Zolgensma. Its indication is for spinal and muscular atrophy. This particular drug is most expensive drug in the world. One injection costs about $2.1 million. And it takes a lot of effort. And I have seen some news in India, some kids, they have it. And it costs so much, cost that people over there can't afford it. Similarly, there are some more drugs like Luxturna for inherited retinal diseases, lipodystrophy. This drug is called Mylept, and so on and so forth. So these drugs are very expensive. It takes a lot of time and effort from various people to bring these drugs to the market. So now, we do need to understand the basics of biology, which is important. So we all know in our body, human cells, there are different types of cells like lung, bone, liver, brain, blood, heart, muscle. And each of these cells, they have different function and they work in coordination with each other. So understanding this function of these cells is equally important. Now, this is a structure of an animal cell. We all know that it has a lot of different organelles like endoplasmic reticulum that is important for transporting materials from one part of the cells to another part. Nucleus that controls the activities of the cell and hereditary material DNA. Vacuoles is found in some animals cells.
Excuse me, sir. Slides are not changing. Excuse me, sir.
That's okay.
Yes. Yeah.
What happened?
Yeah. Yes, sir. Yes.
Slide is not changing.
So now it is. So it has been stopped at first slide.
Is it okay now? This one?
Yes, sir.
Okay. Sorry. So these different organelles in a cell, they perform different functions. Now, how this central dogma of molecular biology is important, how genetic information flows from DNA to protein. So what I'm trying to make everybody, understand in a very simple term, and my focus is for undergraduate, all these young scientists. From gene, which is DNA, its transcription happens, it forms mRNA. Then translation happens, it forms protein. So if protein is normal, it will function normally. But if it is a disease protein, it could be many factors can involve, can cause disease. Now, how do you fix that particular protein which is diseased? So there are different approaches. So what you do, either you have an inhibitor, small molecules, or agonist of protein. We heard yesterday 2 talks very nicely presented by Dr. Satyanarayan and also Alok Ranjan. Both very small molecules. And for the Satyanarayan, for the CRC, the cancer, for Alok Ranjan was for HIV, the compound extracted from the plants, right? So those are the small molecules that people have used. However, there are other approaches, biologics, inhibitor or mimics of protein, right? So these are different approaches people have been using. These are there are so many drugs out there which have been in the market for different disease indications. So here what I'm showing you, I'm not sure whether the full screen is coming. Everybody can see the whole picture?
Yes, sir, it is visible.
Okay. So, because, if I, hold on a second. So basically there are 4 therapeutic platforms. The first is small molecule. Small molecules, you have 2 examples, aspirin and ibuprofen. Protein replacement, you have insulin, which is another one is named like Exubera, inhaled insulin. And monoclonal antibodies. One example is Keytruda for cancer, marketed by Merck. And the 4th platform on which I'm going to talk about is RNA therapeutics. So these 2 drugs, Kinamro, that has been approved, first systemic injected drug, produced by my previous company, Ines Pharmaceutical, for the cholesterol lowering. It happens in some of those patient whose cholesterol cannot go down with the traditional small molecule. And this particular antisense drug called SMA, spinal muscular atrophy, and I will talk a little bit about this one later on. And these 2 drugs are antisense oligonucleotide based, whereas another the compound approved based on siRNA therapeutics is called Onpatro. Onpatro is for a disease called transthyretin. It's a polyneuropathy, is a disease of nervous system. And we all know that COVID vaccines, by Pfizer and Moderna, which are mRNA-based. So as you can see, siRNA-based, mRNA-based, and these compounds, 2 of them are ASO-based. There are several more. I'm just giving a few examples. Now, this particular slide shows the drug discovery and development timeline. So here you can see it takes about 14 to 15 years to develop a FDA-approved drugs, which starts from you screen about 10,000 compounds. It could be a lot more. And then once you identify, narrow down a lead compound, suppose 250 compounds, it goes to preclinical steps. Like, it tests in different mouse models, cell culture and all kind of thing. And then out of 250 compounds, one can take 5 compounds and, do the clinical trials. And then out of 5, one looks best based on safety and efficacy. FDA will approve one drug. Now, the typical drug discovery path here, again, it shows about 10 to 15 years, and also it costs about $2.6 billion, or in that ballpark range. So what it entails? So once you have target selection that involves the role of biotechnology, molecular biology, protein expression, and engineering, followed by lead discovery that is also important, that is automated high throughput screening system. And medicinal chemistry is also very important. Without having a good compound, you cannot do anything. So lead medicinal chemistry plays a pivotal role here, vital role, and that involves lead optimization. What you need to do is the structural biology and computational based drug design and modeling. And then once you have that, you need some in vivo studies. Here you have in vivo, ex vivo pharmacology. You also need to have drug metabolism and pharmacokinetics, where you can see the drug kinetics after injecting in the mice or large animals. And once you have the drug, pharmaceutical science has an early development, and then it goes to the project management and then FDA and approval. In addition to that, there are also role of bioinformatics, structural biology, chemoinformatics, and system biology. The same thing. People also have in silico drug design and development pipeline using docking, QSAR, combinatorial chemistry, pharmacometrics, ADME, all kind of tools and techniques are needed in order to have a good drug approved by FDA. Now, targeting small molecule therapy. So again, this is just one slide I want to show you. It's very similar to what I showed before, is that you have to have a molecular target identification. So that is very important. So suppose some basic science researchers, yesterday we saw published that this particular disease, this particular protein is involved. So that's called molecular target identification for that particular indication. And after that, you identify where this can be expressed and what other tools you can use to establish a assay protocol. And then you go into high throughput screening using a different compounds targeting different region, different compound modifications, so on and so forth. And then these are the main areas of research here. You can have screening hit identification, in vivo efficacy verification, and preclinical candidate declaration. So hit to lead optimization. One of the important aspects of drug discovery development is patent filing. So once you have your ideas, you need to file a patent, because otherwise it's not protected. Anybody can copy your hard work and they can take advantage of that. So this is very important. So usually the patent life, it varies, usually I think 15, 20 years. And after that, it becomes anybody can, make a generic compound out of that. So there are several companies, and because once you have patent, usually these drugs are very expensive, but once the patent is expired, any company can make a generic compound, and that becomes cheaper. And India, China, other countries are doing quite a bit of that. And it also helps patients. Now switching to this main topic for today's presentation is targeting RNA for therapy. So as everybody now knows about Moderna mRNA vaccine, as well as from Pfizer. But the company, I used to work, they are in the business from 1989, and they have been pushing very hard to get a drug out. and they target RNA using a technology called antisense oligonucleotide, which I will just explain a little bit later. So what I'm trying to say here is that RNA is the fourth platform, and it is getting a lot of traction nowadays. And this small, slide portion here shows a variety of RNA. This is, I just pulled together some brochure from the published, media where,
I would like to interrupt a little. Actually, slides थोड़ा सा change नहीं हो रही थी. That is what I was saying.
Now my end is changing. My end is changing.
Yeah, it is okay now, sir. Actually, whenever you speak the next slide, you immediately kindly change that. Yeah, this is now okay, sir.
Okay. Is it okay now?
Yeah, yes, sir. Sorry for interruption.
No, that's fine. I need to, make sure that, what I'm presenting, people are, following up. Otherwise, no point.
Yeah.
Right. Pulled up internet, various. So and microRNA. And then the fourth one is pvRNA in RNA silencing, and that is also involved in epigenetics as well as other small non-coding RNA, such as RNA-SP.
Sorry, sir, the slides are still not changing. The rise of RNA therapeutics slides are seen.
I think there is some problem.
Yes.
I am changing. Why it's not changing?
So you can use slideshow also then.
Okay. Can you see this one?
Yes, sir. Functions of RNA.
Yeah, because I was talking this one, I changed from the previous some. Okay. What happened? See the
Yes, sir, it is.
What I was, let me repeat it. Is a flies. So non says and I may share some slides later on. If not, I will conclude at a certain time. So, disease, there is no cure for this one, but this particular RNA become toxic for the body, and it impairs neuromuscular, causes neuromuscular problem. So you can see over the time, so many RNAs have been evolved, and their function are still being elucidated. So, can you see this slide now?
Yes, sir.
Okay, there is a. All right. So, as I alluded before, that technology, unlike traditional drug platform, they target RNA. So, you have gene, mRNA, translation, and disease-causing protein. And these proteins can be targeted by small molecules or inhibitors or biologics. However, the antisense, which is here red, can bind to mRNA and causes translational arrest, and therefore reduces the protein disease and disease burden. So, can you see this slide?
It's showing antisense technology.
Okay. One. Can you see the inhibitor of antisense?
Get. Yes, sir. No. Antisense inhibitors. Yes.
Okay. Actually, there is animation picture, so I cannot show the animation in this one, but I will try. Okay. All right. So this is antisense inhibitor. This very important slide. One has to understand this particular one. So what is antisense? Antisense, as I previous slide I mentioned, that is a small stretch of DNA that binds to RNA of that particular protein. So what over the time mostly are 20 nucleotide, and they have 5 ways. So one of the modifications here, if you see here, ribose sugar, the backbone is sulfur. And here the O group has methoxyethyl modification. And if you put this modification, and so this is what it is. This is a whole sequence about 5, 10, 5 DNA, and the 5 is called methoxyethyl modification in the 5 prime, and then also in the 3 prime. So 5 nucleotide here, 5 nucleotide here, they have been modified. Whereas in the gap, it's all DNA. So this particular gap is important. So why is this important? What happens when you have antisense here? Can you see this particular small picture cartoon here? Let me see. Let me make it bigger actually. So, is it visible now? Better?
Little bit.
Okay. So this is it gets into the nucleus and it binds to RNA. It produces. Thus, a very important RNA is H. Endonuclease cleavage, and what it does, it binds to that duplex. Endonuclease RNase H cleaves RNA, and then what happens? There is no RNA, no protein. That's why this is. There are more than 6,000 old information.
Showing antisense inhibitor.
Okay, that's fine. I think there is some issue with the. So, for the picture was taken from one of Stanley Crook is a doctor, scientist, everything. Particular, slide. What I'm trying to make everybody understand that there are various, ways this antisense mechanism can work to modulate gene expression. The first one is enzymatic RNA degradation, and the second, so enzymatic RNA degradation includes what happens that mRNA is formed. So this A RNase H1 that binds RNA, it can bind to exon, intron, anywhere, and then it causes RNase H to come and degrades the RNA. And same thing in the site modulation. So this particular one, it does not degrade, but it changes the splicing pattern of the. Suppose in some cases, I will show one example, nusinergen, the one I showed you, the drug Spinraza, is changes the splicing. What happens sometimes there is a missplice. As a result of that missplicing, there is a protein, truncated protein is formed, and which is non-functional or less functional. So that particular splice is important too. That is corrected by one of those antisense molecules. And then there is a occupancy mechanism. Other one is modulation using non-DNA-like antisense, like this particular one. And then other one like translation activation that inhibits the translation by steric blocking. And then this E1 is microRNA, modulate the microRNA to inhibit function, base pairing with the microRNA binding of a to a microRNA. So there are various mechanisms that can be utilized using various state or compartments. So this picture shows distribution of the and the label type. Found this very, very small, but what I can tell you that most of the drug cortex and very, very small in different other organs. So this that is small to screen to come up with the one compound as a lead and the approval. But here, this technology allows that even like you have 3,000 or any other one. So from and then which a point is an issue, whereas with the highly specific, and as I'm one of the in my that Aruimaji mentioned in my introduction that the modification is very important. That increases the stability and efficacy of the compound. But over the time, our chemists have done marvelous job in modifying various places in this particular, ribose moiety and other linkers. So like R here could be H if you're DNA, or hydroxy group, it could be RNA. So 2' substitution here is very important for sugar you have, here for the sugar. And also you can conjugate this with different, entity, base modification. So suppose 2 fluoro modification fluoro here, that improves the binding affinity of target RNA, right? You can also have 2' O-methyl modification here at this place, improves the PK, means, the drug is nicely distributed in the body, and also modest improve the potency. 5-methylcytosine at this place improves the affinity of the RNA. This one is also very popular, 2'-methoxyethyl modification here at this particular ribose. Now, there's PS backbone chemistry here, which is like phosphodiester. You can have sulfur here to make more stable. Otherwise, this phosphodiester bond can be degraded very easily. So you have PS backbone. So people do change PSPO backbone to test which particular combination is better. Sometimes if you have like 10 nucleotides, you can have 5 POs, 5 PS backbones, and other modifications. And then thiophosphoramide, there's so many bunch of modifications. Another one is constant ethyl modification here on the right side in the green. So green, what you see here, that this particular constant modification is very, very good. Actually, increases potency and half-life to 2 to 4 weeks. Another modification is log nucleic acid, LNA, that improves the binding affinity. So you can see there's so much modification has been, taken place. But in addition to that, there are other modifications, which is, the delivery, to improve the delivery of these compounds. Can you see this slide, please? Let me know.
So it is showing oligonucleotide modifications.
Continued? Okay, hold on a second. Okay, now, can you see this one now, colored one?
Yes, sir, continued one, yes. Okay,
so now, this particular one, you have, for the delivery, the key formulation and conjugation strategy to late stage clinical trials to enhance delivery, lipid nanoparticles. Also, you can have, GalNAc conjugation, is targeting for the liver, different fatty and, lipid conjugation. You can conjugate that oligonucleotide with this and improve the, delivery, and also reduce the, doses and less frequent. And on the right side here, there are single and double stranded oligonucleotide. You can see single nucleotide here. The green denotes the nucleobases, oxygen is red, and yellow is sulfur, and the orange is phosphorus, gray is sugar. So there are different modifications. This is the nice structure, so I thought to share with you. This is single stranded, oligonucleotide. This is siRNA double stranded. So now, as you can see with the time, how the medicinal chemistry is improving the anti-seizure technology, like it's called generation 2, methoxy modification, and then, over the time, it has become a generation 2.5, and then you have conjugation. You can conjugate generation 2 with it's called ligand. I'm forgetting this particular abbreviation, but it's sort of conjugation. Different conjugation is a common term for generation 2, and also you can conjugate generation 2.5. And all they do, it increase potency, decrease dosing volume, and they target different tissues. Okay, now next one. Now, can you see this slide?
Yes, sir.
Okay, so now I will show you this particular slide. So these are the tables for commercialized RNA targeted drugs. As you can see, there are a whole bunch of them. This is a partial list, but what I'm trying to show you is Spinraza. This is a blockbuster drug for spinal muscular atrophy. And for this, these two people have been, this is called, breakthrough prize in the life science at 2019. And I was fortunate to work with Dr. Bennett. And actually, this particular prize is called Industrial, Nobel Prize. And this is such a, important discovery happened at INIS. And, a lot of, children, who cannot survive beyond age 2 are benefiting right now. So what is muscular atrophy? It is like, it's a rare disease. And so can you see this slide?
Yes, sir. Spinal muscular atrophy.
Okay, good. So this is like, 6,000 to 10,000 children in US. And, usually the SMA inherits 2 copies of a missing or faulty gene. And there is a protein called survival motor neuron 1, SM1 gene. So in this particular, in these children, the SM1, SM1 protein is not made because there is exon 7, is missing, right? But there's a unique technique that antisense, allows to make this correct. So I will show you very quickly. So now, just briefly, there is type 1 SMA. There are 60% of people have type 1, type 2 intermediate, and type 3 and type 4. So these type 1 is most common, and children, they have respiratory infections, collapsed lung, and they don't, they survive before their second birthday. Okay. So this is very, very important that, a drug has to be made. So what I'm going to show you, let's focus on this particular, diagram. Can you see this one? So what happened that, so this is so nice that nature has made SMN1, SMN2, the multiple copies, and the protein that is, also has so the protein encoded by SMN1, 2, 3, 4, all function, but they have different copies. But what happened that in SMN1, the exon is that particular missing, but in SMN2, that particular exon, you can treat and skip the exon with the antisense, and that allows increased inclusion of exon 7. That allows to form a full-length SMN protein, and that is helpful for the kids in all different types. So this is what it is. So what happened that it binds this particular compound, which is it's not more gapmer, it's like a straight 2' ethoxymethyl compound, and that it binds after in this intronic region at the junction here, and that allows to inclusion of exon 7, and that forms more full length this protein. And then but there is very, very small amount of this particular truncated protein is left. So what I'm trying to say that this particular technology has allowed to help these patients to improve their life. Otherwise they will die. So there was no other tool. But now in the beginning of the slide, when I showed the list of all these different drugs, Joel Jasma and all, there's another one for SMA that came later on than this Nusinergen, which is also known as Spinraza. And these drugs are somehow expensive, and the way it is delivered is delivered through intrathecal, in between your vertebrae. So what are the advantages of antisense platform for drug discovery? So it's rapid identification of drugs, efficiently screen many targets in parallel, 100% success rate in identifying inhibitors. All genes are druggable with high selectivity. You can predict the pharmacokinetics and safety. You can share manufacturing and analytical processes, and shorten the timelines from concept to first human dose. So this is a more or less similar information that I presented before. Now this is very interesting, study. You may have heard about, personalized medicine. So there is a rare genetic disease called Batten disease, where it's a neuromuscular disease where there's a mutation in the gene called CLN7. So what these scientists have done, they have customized this oligonucleotide therapy, first of its kind in the world, for just one patient. So this particular girl, her name is Mila, and that's what they name the drug as Milasen. Okay, so what this gene is CLN disease, and it has been the oligonucleotide they customize in a record time. I will show you the timeline in a short while. So CLN7 stands for neuronal ceroid lipofuscinosis, and also is called MFSD8 gene. So when this particular girl was born, she had a lot of issues. So let me go back. So here is the presentation. So when she was presented again to the clinic, she was 6 years old at that time. And she had like blindness, ataxia, seizure, developmental regression. And her right foot began to turn inward. And at age 4, she will, pulling books close to her face at bedtime. 5 years, she came to medical attention. And, a lot of issues started happening. And their biopsy, what they found is that there was abnormal electron-dense lysosomal inclusion in the swirling fingerprint pattern. And there's a lot of other neuronal issues like MRI. They reveal that mild cerebral and cerebral atrophy, and EEG reveal that they are subclinical generalized seizure. And this particular is a disease autosomal recessive, and it has mutation, single pathogenic mutation G to C in gene MFSD8. And coming back here to the previous slide, that this particular drug is called milasense. It's a splice modulating antisense oligonucleotide. Is tailored for a particular patient. The cell line from the patient serve as a basis of launching N of 1. And usually when you have clinical trials, N is equal to could be 2,000, 3,000, but here just one patient, it was just N of 1. So within a year, they contacted the patient and drug was developed very, very fast. So this particular disease is aggregate affect approximately 30 million people in the United States. And next generation sequencing has really revolutionized their diagnosis. And it is really, really helpful with all these new tools. So let's go back and next in this slide, let's see if I can you see this particular slide? Let me make it bigger. Okay, so study timeline and genetic diagnosis. So when Mila was, came in the clinic, I think it's Boston. She's from Colorado, but she came to Boston. Clinical diagnosis happened in 2016. Everybody can see this slide, study timeline, genetic diagnosis?
Yes, sir.
Okay. And then it was a referral came here, and they identified the mutation in early 2017. And then they established a patient cell line. And then they have identification of a splice defect. And after that, they identified the ASO in September, in middle of September, October. And then they have validation of ASO functionality somewhere, October and November. And then in parallel, they were running the tox study in rats. And with the FDA, permission to proceed, and first drug dosing happens in February, late, January, early, here, just at the late February actually. So you can see from 2016 late to 2017, about 1.5 years, you are already here. Such a fast way of developing a drug, as you have seen with mRNA COVID vaccine as well. How fast that drug was developed by these scientists, because this mRNA and RNA in general, this is a new platform that is really impacting human life and helping patients. Now let's go back and see this left panel here. This is the patient's whole genome sequencing realignment near MFSD intron here. Okay, intron 6. Okay, now let's focus on this panel C here. So right panel, what happens that in this particular disease, there is a retrotransposon that got usually, we have heard, learned, early on in college or, early high school that, retrotransposon, they jump in the genes, right? So what happens in this case also, what happened that they found that SB1, this transposon just got inserted, after exon 6. And then it just, after the intron 6, it caused like a premature stop codon here. So premature stop codon, it will not allow to the genes to transcribe, and therefore you will have a truncated protein. So that's what they found. And the protein was in oligonucleotide drug development. oligos, they can see there is a increased number of exons 6 and 7. So that means it is helping making more protein, right? And then they analyze this RNA sequencing validation here on the left panel. See here, you can see with the mock, you have 8% of exon 6 and 7 splicing. In the scramble oligo is like, it's like a analog, is inactive part of the active antisense. It has about 11%, whereas in the MILA-sense TY777, you have a 34%, which is remarkable. Now, this is very interesting, this particular panel here. Let me make it big so that you can see. So the hallmark of this particular, in the disease, there is vacuole formation. So this, here, the patient fibroblasts, they have a lot of vacuoles, and the control fibroblasts come from the non-patient, which is, there's no untreated, there's no vacuole. Now, when you treat with the scramble oligos, so the score 5, they have scored this vacuoles, like 0 to 5. 5 is the maximum number of vacuoles, 0 is no. So 5, you have patient fibroblasts, lot of vacuoles. 0, this untreated control fibroblasts, you don't have anything. And then in the score 4 is for the fibroblast patient, which are treated with the scramble oligo. Where in the MILA-sense, you can see the score has gone to 2. Huge improvement. And then, the body start making that protein and also improving the function of that particular, patient. Now, can you see this slide? N of 1 clinical study?
Yes, sir.
Okay. Now, let's focus on this one, that this is dosing schedule. So what happens at dosing schedule, you dose in the milligram, different doses, 3.5, 7 at different time. It's called escalating loading dose. Like, you increase the dose at different days, and then there is maintenance dose. You keep the same dose for, longer period of time. So this is how this, dosing schedule works in particular, this particular, setting. So since this drug is given intrathecal, similar to, Spinraza that I alluded before, and then you have to measure how much drug is in CSF, is cerebrospinal fluid. So what they can see, with the time, there is a small, increase, and then it goes down. But after continuous sustained, dosing, you can see that the drug MILA-sense is continuously present in the CSF. And then they measure the seizure, because this is another hallmark, that seizure frequency over the time, you can see the frequency and the duration recorded by the parents is keep on going down and down and down, right? And now the lower panel here, the left panel, they did the seizure count. So on the left side here, the seizure per 24 hour, where is increasing dose here. So if you dose, it's a red star here, you can increase in dose, you can see decrease in the seizure count, and then it comes back again. But it's working so well. And then they also see there is a 52% reduction in seizure duration. You see here, duration, like, used to, like, how long a person is under seizure, like more than 2 minutes, but here she can go less than 1 or 2 minutes, right? So with the increasing dose, you have like a lower mean seizure duration, and then it comes back and then goes back again. And now you have percentage time is spent in seizure. So how much time Mila is spent in particular seizure time. So it's a cumulative time is spent about 85%. So usually before the drug, she will go more than 2 minutes and multiple times in a day, about 15-20 times. But with the drug, the seizure time reduced and frequency reduced both. So this is very remarkable, like, outcome that, such a short time, this particular, is a patient-centric, drug made, and, she was able to got benefit. And, I'm almost there. So with that, I think I don't know what is the status of this particular girl right now, but this what it proves that it's a very powerful technology that can be used in a very short time, for different rare diseases where small molecule biologics and other, approaches cannot work. Now, the summary and conclusion. So, can you see this slide?
Yes, sir.
Okay. Am I on time?
Yes, sir.
Okay. So, RNA-targeted therapies represent a platform for drug discovery and development involving chemically modified oligonucleotides targeting a wide range of cellular RNA. There are several RNA-targeted therapies are approved for commercial use for indications as diverse as spinal muscular atrophy and reduction of low-density lipoproteins and cholesterol. Various routes of administration, including subcutaneous, intravitreal, intrathecal delivery, has been well documented. This technology is efficient and supports approaching undruggable targets. Progress in understanding the molecular mechanism by which these drugs work has led to steadily better clinical performance and a wide range of mechanisms that may be exploited for therapeutic purposes. So in conclusion, targeting RNA is a viable option by antisense oligonucleotide. So an siRNA, another antisense approach for drug development and eventually impacting human lives. Now, can you see this slide, KG Publisher Books?
Yes, sir.
Okay, so these are some of the books like I just mentioned before that my father wrote, and these medicinal chemistry books that are very popular in pharmacy schools, but there are books for organic chemistry for undergraduate graduate schools. Sorry. I'm just taking this opportunity to share these books because it's chemistry department, Navyuga Mahavidyalaya. And if anybody interested, to get more information, you can contact Rajesh Pandey in Banaras with his mobile. With that, I thank you everybody for listening to my presentation. I think I was not expecting this kind of issue in my presentation, but what can I do? Anyway, so this is. So I conclude my presentation. I'm on time or?
Yes, sir, you're on time. Thank you, Dr. Sanjay, for a wonderful and inquisitive talk on RNA-based therapies. I'm sure our participants gets benefited from this talk. If anyone has any query, he or she can ask.
Yes, ma'am. Myself, HN Singh.
Yes, sir.
My one, not query, the request to sir. सर कल भी मैं कई लोगों के बड़े नोबल लेक्चर सुने थे, आपका भी नोबल लेक्चर जो है केमिस्ट्री है, तो व्हाट टू स्टडी तो बहुत मिलता है, बट हाउ टू स्टडी सो दैट इन ए लेस एफर्ट वी कैन अप्रिशिएट द फिलॉसफी ऑफ द सब्जेक्ट मैटर. तो सर इसमें जरा आप लोग जो है ना कुछ
I don't know what the issue is here now. I can see. Let's see. New window. Okay, here. Let's see here. Can you see the slides now?
Yes, sir.
Okay, let me go back here. So the toxic RNA, I will show you just one slide here. Okay, two actually. And can you see this slide?
सर दिख तो रही है बट लेकिन बहुत शॉर्ट छोटी दिख रही है.
एक मिनट, अब दिख रही है?
Yes sir.
Yes, sir. It is visible. Yes, sir.
Okay. So hold on a second. So this particular disease, I will just take 2 minutes. So I don't want to go overboard. So this particular disease, was first described by a German scientist in 1901, and hundreds of years there was no information available until I just stumbled upon this. My company, I just started working with the collaboration, and then in 2015, we were able to get a lot of work done, but still work is going on. So what this toxic RNA is, hold on a second. So here is example, okay? So what happens that, let me make it bigger. Can you see this? Let me see, make it bigger.
Sushmita ma'am, mute yourself.
Yeah, somebody, okay. So can you see this slide?
Yes, sir.
Yes, sir.
Okay. Okay. So now what happens that, this is called, toxic RNA, because what happens that in this DNA, when there is like, trinucleotide in this particular patient, population, what happened that, most of these patients are found in Canada.
Sushmita ma'am, kindly mute yourself.
So this particular, DNA, on the 3' end of the DNA, there is CTG. It's like CTG, keep on adding. So what
Sunil ji, kindly mute from your end. Yes, sir.
Okay, so as you can see, so what happened that this particular gene is called DMPK gene, dystrophy myotonica protein kinase. So DNA, there is CTG repeat expansion. So in the nucleus, it gets expanded and it cannot come out of the nucleus. So what happens that it start making a loop, and then it sequesters proteins that are important for the function, and then it is not available. Then the problem happens. So with the drug, what happens? So this RNA becomes toxic. So any DNA with the expansion, it will form an RNA, and then that will have that particular repeat correspondingly. So it will not function. It just derails its function, and then these people cannot walk properly. And also there is a very unique thing. So interbreeding is a key. Because if suppose somebody is identified as age 70 this disease, their children, next generation will be 40s, third generation will be 20s, fourth generation will be, what do you call, in childbirth, like 2 years, 3 years. So the expansion is usually 200 CTG repeats, and the next generation it becomes like thousands of repeats. So it's become very congregated. So the symptoms appears very early. So with this technology, we were able to publish a work on this particular one, and actually this was a pretty neat study. When we published this particular paper, that was like highlight of the here this Nature paper. It was like they wrote a review, and they said this is a breakthrough in the genetics. The reviewer wrote. So that time it was not known that how to target this disease. So this particular RNA that called quote unquote toxic, because it's not doing any good compared to what other functions I mentioned about RNA, that are like a protein function and other thing, right? So that's why they call this a toxic RNA. Am I clear?
Yes, sir. It's clear.
Okay.
So presently कौन से drugs use हो रहे हैं इस therapy को use करते हुए?
अभी इसका कोई ड्रग नहीं है. Actually हाँ, आपके इंडिया में बहुत से पेशेंट्स हैं. इंडिया में बहुत से लोग जाते हैं न्यूरोमस्कुलर, उनको होता है प्रॉब्लम, but they do not understand कि वो क्या हो रहा है. मैं तो जाता हूँ इंडिया में, कई जगह टॉक देता हूँ, लोगों से बात करता हूँ, लेकिन चीजें अनफॉर्चुनेटली बड़ी स्लो होती है रिस्पॉन्स में. बहुत किसी को कुछ हुआ तो अरे भाई ये हो गया, तो चीज doesn't move properly. इसी से मैं बहुत ही emphasize कर रहा हूँ इंडिया में RNA based drug discovery के लिए people should emphasize. It's a huge market. Not only that, to learn, we have a lot of good chemists, we have a lot of pool of good scientists. मैं तो जब भी जाता हूँ तो I just try to address this. But I'm working on hoping someday we'll get this thing rolling. RNA you can study, you can publish paper, but how to improve the clinical manifestation? These are the new areas we should encourage students to come forward and learn.
Thank you, sir.
Okay.
Excuse me. Question.
Sure.
Yes, sir. Sir, in spinal muscular atrophy, the technology is used that just increase the lifespan of that children, or also ends completely the disease from them.
So you are mute.
Okay, could you please repeat the question? I'm sorry.
Yes, sir. In spinal muscular atrophy, the technology is made that only increase the lifespan of the children in which the disease is found, or completely ends the disease from them.
As the disease from them?
सर मतलब वो पूछ रही हैं कि वो जो ये थेरेपीज हैं उससे वो बीमारी पूरी तरह ठीक हो जाती है या मतलब उनका लाइफ स्पैन बढ़ जाता है, मतलब वो
नहीं लाइफ एग्जैक्टली तो लाइफ स्पेन उनका बढ़ जाता है. यूजुअली उनका लाइफ स्पेन दे डाई 2-3 years old. They cannot complete, but now even after 7-8 years, it was approved in 2016. So all those the kids are still surviving and their life has been increased for sure. So those kids are still being followed upon, right? Let me put it this way. So there are 2-3 more drugs to treat SMA.
Okay.
Yeah. Thank you, sir.
You welcome.
Sir, one more question. Sir, wanna listen to your journey as a student to a scientist.
Yes, of course. We all have to work hard, the first thing, and also you have to follow your passion. Growing up, in academic area, my sister, I think she's audience, she's also a chemistry PhD. And, our parents, instill these things, and we actually, you have to push yourself what you want to do in future and work hard towards that. Talk to people, interact, networking, get some, information, and I will be happy. Email is there, if you want any question, I will be happy to answer. This is what it is. I'm just trying to devote my time to, help children. They want to learn something, sure. That's what my father used to do.
Thanks a lot, sir. Thank you very much.
Thank you, Husna, for the question. Thank you, sir. Now it's time for our next deliberation, which is going to be presented by Dr. Upendra Rao Gola. And it is our great privilege and honor for me to welcome Dr. Upendra Rao Gola, who is presently working as postdoctoral fellow in Department of Medicine, Penn State College of Medicine, Hershey, Pennsylvania, USA. He had obtained M.Pharma degree from Jawaharlal Nehru Technical University, Hyderabad. He pursued PhD from Indian Institute of Science Education.
Is it visible now?
Yes.
Hi everyone. Today I will be talking on the emergence of green technology, nanotechnologies for the sustainable environment. So basically I am not belonging to this exact field, but I will be more familiar with the nanotechnology and the use of its nanotechnology for the biological implications. And today I will be talking a little bit of its broader implications and the challenges for the sustainable environment applications. So today I will be talking about what is nanotechnology and then what is the green technologies and current status of our Earth in terms of environment and why we need the green nanotechnologies and few examples of green technologies for sustainable environment along with individual responsibilities to protect the environment and finally I will conclude my talk. So first I will try to introduce the term like nano and the technologies. So a nanometer is a unit of length in the metric system that is equivalent to 1 billionth of meter, like 10 to the power of -9. Like it's a very smaller unit of a metric system. And it comes to the technology, it is like making usage and knowledge of tools, machines and techniques in order to solve a problem or perform a specific function. So when it comes to the nanoscience, it is a study of phenomena and manipulation of materials at atomic, molecular and macromolecular scales where properties differ significantly from those at a larger scale. And the nanotechnologies are the design, characterizations, production and application of structures, devices and systems by controlling the shape and size at nanometer scale.
Slides are not changing, sis.
Not changing?
Yes, sir. Yes, sir. Characterizing nanomaterials.
Oh, okay. I think I have to no presentation mode.
Yes.
Okay. So today, when it comes to the nanomaterials, like as I mentioned, it's a very small scale objectives. And so here we have to characterize whether it's a nanoscale or not, like to get a proper size and shape estimations. So of an aerosols, nanotubes, nanoparticles. So everything like more of nanostructures need a proper characterizations to whether they really achieve the nano size or not. So generally, it maybe depends on the origin of materials and the chemical composition that were used, and the aggregation state of those particles, whether they are single particles or they aggregated to larger molecules, they make a bigger size. And sometimes the shape of the nanoparticles, like nano, should be properly characterized, whether it's a spheres or needles, like platelets and tubes, based on the interest. And also we should look at modifications, whether it's a treated or coated or non-treated, like those properties also should be very well characterized. And also the dispersions also like should be very well characterized, whether they are gases or like some aerosol. Solid state should be very properly confirmed. Like nanotechnology is defined. So green nanotechnology is a branch of green technology that utilizes the concepts of green chemistry and green engineering. It reduces the use of energy and fuel by using less material and renewable inputs wherever possible. So nowadays, there is a tremendous amount of research with potential applications in the different industrial and analytical fields, as well as clinical application for diagnosis, treatment, and illness and drug discovery and delivery. So because of that, the search for the biocompatible and toxin-free nanomaterials is mandatory to move from the bench to the real world and the safe use of these materials in the nanomedicine. And it is also known as environmental technology and clean technology because it is not going to harm the environment in any aspect. So the applications of green technology like varies from different specializations, can be used in bioengineering and nanofabrications, cosmetics, and energy utilizations and medicines and drug discovery, and nanobiotechnology for the delivery systems and optical engineerings. And when it comes to the green nanotechnology, there is a newer developments from the last 1 or 2 years. The use of phytoformulations for making nano particles, that is making it's more eco-friendly and it's more safer for using those phytoformulation derived nanoparticles in the biological usage. So the green nanotechnology, like in phytoformulation, significantly contributes to environmental sustainability through the production of nanomaterials and nanoproducts without causing harm to human health or the environment. Metal oxide nanoparticles such as gold and copper oxide, and zinc oxide are very important and have been synthesized from number of plant extracts without causing a harm to human health or environment. So this is one of the recently developed approach where the plant-based extracts can be used in combination with metal ionic sources to make them nano-sized materials using the nucleation approach, accumulation, and then making a stable and capped nanoparticles through these phyto-based extracts. These are more eco-friendly than the previous chemicals. And also these biological synthesis and applications of these metal nanoparticles through using plant-based products have a biomedical and environmental field effects. So these can be like the nano size can be achieved using plant-based extract as indicated, or else like can be used as smaller microorganisms such as budding yeast and bacterial cultures or fungal cultures can be used to reduce the size of those materials to the metal ions to make a nanoparticles. And the biological synthesis of these nanoparticles have wider applications in field of like antimicrobial activities, as I mentioned, cosmetics, and then photoimaging purpose, and photothermal therapies, and magnetically responsible drug delivery systems, and various type of human cancer treatments also can be targeted with these metal nanoparticles. And the nanosensors also can be detect the biological molecules and environmental factors. So when it comes to the green technologies, we know that technology should help to achieve high quality life for more people on the planet, and it should not drive any of the just in the market by the analysis of trends and vision of the preferable future. But the policymakers should create a framework for innovation and technology development based on their view of the future. And also the technology should respect environment and ecological conservations of otherwise they will not be sustainable in the market for a longer time. And all the technological innovations should take our ecological limits into consideration before the development and then implementation to the application purpose. So what is the sustainability? So in terms of sustainability, like it can be a development that meets the needs of the present without compromising the ability of the future generations to meet their own needs. Like we should be having them for the next generations for the usage of them. Like, so we should not end the products. And the more goals of these green technologies are like, we should be able to rethinking to use a proper technology and proper use of the ideas to make it a more of applicable to the society. And the recycling, like most of the products that we're going to be using into the making the formulations or something like can be try to be a recyclable ability should be tested. And also renewing, like something like whatever we have, the old materials can be generated into recycled and then making a new product out of it, that is renewing the old items. And also the reducing them, like we should reduce the contaminants and we should reduce the pollution impact of those materials that will be used in the technologies. And also one should be very responsible before use of any technology for the impact on the society as well as the market. So generally, when it comes to the basic needs of our living things, we all know like any living things on the earth need proper food and proper water, healthier water and proper air and sunlight and shelter and the soil in order to make a good food out of the soils. So these are the basic needs. And when it comes to the current state of these basic utilities that human beings are exposed now, so there is a lot of, when it comes to the change, like sunlight utilizations. So nanocrystal solar cells, this is a nano-derived solar cells. We all know solar cells that are very cost effective to convert the sunlight into energy, but there is a modulation of the solar cells by incorporation of nanocrystals to the solar cells, making it a more and more effective conservation of energy and storage utilization. So here we use the basic structure of nanocrystal solar cells. They use a highly conductive, low-cost aluminum foil as a substrate bottom that make the electrode of the cells, and it has a printing of those CIGS called like copper, indium, gallium, and selenium semiconductor materials as a ultra-thin nanoparticle that inks the minimizes our skin for the expensive high vacuum manufacturing equipment can be negotiated here. And the nanocrystals are typically based on silicon and cadmium telluride or CIGS based substrates, or generally silicon or various organic conductors are used for developing the nanocrystal solar cells. And when it comes to the efficiency, the normal solar cells and the nanocrystal solar cells are very outperformed in comparison to the harvesting of the solar energy. And it comes to temperature changes, like we all know the Earth is changing and we have a global warming phenomena where we have temperature is raising every individual year, like by every year. So this is the recent very thermal scan of the world where it is showing there is a tremendous increase in the temperature like of the Earth, like from the last decade, like 2011 to 2021. There is a tremendous increase from the baseline from the 1950s. There is a changes in the global warming effect, and the main activity, like main reason for this global warming, is when it computed to the global surface temperature changes, the human and natural drivers are very contributing to the highest changes compared to natural drivers alone. So the human activities that we do are impacting the Earth. Main features like soil and water and air, everything is changing and making it global warming effect, leading to the higher temperatures. And also the carbon dioxide emissions from previous year, it is every time it's going high and high for the each year, and it comes to the accumulation of those gas usage and oil combustion and coal combustions, and the land usage will lead to the highest amount of carbon dioxide emissions as of now, leading to this global warming phenotype. So when it comes to the pictures of here, like I will show 100 millions of Indians live in areas with poor water quality. So both surface water and groundwater resources are polluted, and due to reduced wastewater treatment here. And the climate changes will increase threats to water security in the recent years coming expected, and the magnitude of floods and droughts also lead to further pollutions of those groundwaters. And with the national supply predicted to fall 50% below by within the next 2030, in the next 20 years. So as you can see, like there are main following reasons are like industrial waste and improper practices in the agricultural sector, and reduction of water quantity in rivers, in plants, and social and religious practices like dumping dead bodies in water, bathing and throwing waste in the water, and oil leaks, acid rains, and global warming, electrification, and inadequate industrial treatment of waste, and denitrification. So several processes that human activities will lead to water pollutions in India. And if you look at the holy Ganga, like it's not a holy at all, like no one will able to now take a bath deep into the holy river as of now, because it's so much polluted, and it's not healthy for current treatment. So we need proper technologies to clean up those water contaminations. So here it comes to the use of nanotechnologies for the use of treatment of water, using those two approaches. Like there are two approaches that can be used. One is a pump and treat system, where we dig the water out from the ground and water treatment plant with using iron-based nanomaterials, and then again injecting back into the water, like into the ground, to the injection well. So this is like a circulatory system where take the polluted water and then clean up and then again re-inject into the earth underground. And there is another approach where groundwater flow, where water is flowing from one area to another. So there will be can be installed that permeable reactive barriers can be installed, so that contaminated water can be purified again, and then making it a pure water, like using based on the ferritin based material or iron based materials here. So this is one of the approach that was tested and found effective for the groundwater pollution treatment using the nano based material. And there are a lot of advancements that are coming up for the treatment of those water pollutions. So this is one of the recent method that was developed in 1 or 2 years ago, where they use a high performance capacitive deionization, disinfection of water with graphene oxide, graft quaternized chitosan based nano hybrid electrode coating. So basically they have made a nano material here where they have combined graphene oxide with quaternized chitosan to make a nano-based formulation, nano hybrid. And this one is like coated with the different electrode-based material, leading to the killing of those bacteria which are flowing like from this tubing. So bacteria infested water is passed through one end to another end, and while applying the voltage here, it is able to kill the live bacteria due to high voltages for the electrodes, and the pure water is coming out from this hybrid, like nano hybrid material. So based on the initial findings, it was found very effective, and the electrode can achieve almost 99.99% killing of those E. coli bacteria in the biocontaminated water that was flowing continuously through the CDID cell here. So this one, this nano hybrid material offers an economical method towards ultra-fast, contaminant-free, continuous killing of bacteria in biocontaminated water. And this proposed strategy can introduce a green in situ disinfectant approach for water purifications. And when it comes to the air, like we have looked at the water status now, and when it comes to the air quality also, we are one of the third highest contaminated air we have, like in India. And as of now, very recent scan in 2020, we had a lot of air pollution that was responsible for 160,000 deaths in the world, and it can be increase the likelihood of dying from COVID-19, asthma, and stroke because of the dirty air that we are breathing every day. And the sources of air pollution can be like dust and constructions are majorly counting for the major air pollution, and there is a waste burning, and then transport-based air pollution, and then diesel burning and industries, and then domestic cooking also lead to contribution to the air pollutions. And the main reasons for concern are like if you are breathing a dirty air due to the air pollutions, the increase of asthma and then the breathlessness is increasing like around 57%. And it comes to the lung infections and TB and general health problems are contributing to the major one. And there is a cancer development, eye infections, and global warming, and there's a decreased lifespan with this air pollution. So one should be very careful, and then use of the technologies to properly use of those nanotechnologies for the clean air. So there is a nanotechnology use for the sensors also. So the nanotechnology based sensors and detectors of pollutions are developed where they have a heavy cantilever sensors where the binding of those molecules like that are present in the air leading to the bending of those chip little bit, and then based on the bending in the lever, it will detect the levels of contaminants present in the air. So this kind of very sensitive technologies using the cantilever sensors are very applicative for the eco-friendly sensors, and also its sensitivity of the contamination and pollution detection is very high with these nano-based sensors. And in addition to the using of these sensors, there is a use of these nano-based products, nanotechnologies for the ambient air purification were tested and found very effective in different parts of the world. So I will be talking about a few of the examples where nanofibers or nanotubes and nanoparticles and nanosheets and 3 different formulations of these nanoscale products are used for air purification using 5 different methods. One is called photocatalysis, where they use the highly reactive molecules for making a highly reactive pollutants to harmless products. And there is an adsorption based, where the active carbons can be used to attract those gas molecules and then make them inactive. And there is a filtration based nano filters, where the aerosol particles can be filtered through nano filters, make the air pure. And also there is a disinfection based metals like gold and other bacterial will be killed from the air which are present in the contaminated air. And there is a room temperature catalysis, where we use a different surface molecules that are reactive towards the air contaminants to make them inactive. So this is one of the example where recently they use the transparent air filters were used at one of the area just to test the efficacy of filters for neutralizing those toxic gases present in the air. And also there is a conversion of those nanofiber film into a face mask was very easily converted and then found effective for the use of those air filtration purpose. And there is as I mentioned, there is a air purification technology with photocatalysis. So basically the photocatalysis is an advanced oxidation process where it involves the irradiation of semiconductors and causes not only the rate but also direction of chemical reaction to be altered here. So once they have a photocatalysis surface here, so when excited with the charge carriers like such as sunlight or anything like photoactivation in the sense like the UV radiation makes them like more active, and then it makes the generation of pre-absorbed H2O and then oxygen to produce reactive oxygen species, which are highly reactive superoxide radicals, which are going to react with the pollutants and then make them non-toxic byproducts after activation of the photocatalysis. And these radicals are extremely found to be strong oxidative potentials and also have a selectivity towards the pollutants, thereby a complete conversion of the gaseous pollutants into a harmless product. So these are the different photocombustion products that are majorly contributing to the air pollution, such as particulate matter and nitrogen oxides and volatile and organic compounds from different reactions that we are causing to the development of air pollution. And there are different photocatalysis agents that are used in the nanoscale products for the air purification method. And there are still like a lot of more agents to be tested and then applicable for the human use in recent years. So when it comes to the so one more, I think many of you know or not, but there are electric vehicles are now in the market and they are gaining the boom because they think that the electric vehicles are green in nature, like they are not using the gas and they are not producing any smoke, but they seems like they are very eco-friendly. But the other fact, like the batteries, like for the electric usage, the vehicle uses like battery cells and body steel and battery housing, electronics and the tires and wheels, rest of the other materials. So they all have a use of carbon emissions to make an electric vehicle are like very high, and it's not completely carbon emission free vehicles. So we cannot say it is a completely green going to electric vehicles, but the impact of gas vehicles as well as electric vehicles when compared, so the electric vehicles found to be a lesser carbon emission in terms of pollution, but they have a little bit betterment compared to gas vehicles. So there is a recent statement about those reducing the battery-based pollution of those electric cars. So the solar roof and the power wall, so they have plans for using the solar energy, like utilizing the solar energy to make conversion into electric energy to charge the cars. And also there is a power wall new tool that is going to harvest the energy and then store the energy for future usage. So there are still like a lot of developments to make it more eco-friendly and then going green in the recent years. So the final eco-industrial revolution should be made. So it comes to the we should respect the planet's ecological limits, and we should recognize the economy as a subsystem of the global ecology and energy system, and we should redesign the products and system and business models copying the nature's functionalities, and we should have a waste loop loops. And also the technology and innovation will play a very key role in the transformation of this eco-industrial revolution. And also the governments will have to set up the frameworks for the eco-innovation revolution. And the eco-innovation is the creation of novel and competitively priced goods, processes, systems, and procedures designed to satisfy the human needs and provide a better quality of life to everyone with a life cycle, minimal use of natural resources per unit output. So relying on the traditional environmental technologies is just not enough. So we should adapt our economies to the carrying capacity of our planet. so that we should make more and more green technologies to come up in the future. And the current challenges that we have with the green nanotechnology is that most of the nanomaterials that have a great impacts on life sciences, however, but they have a inducible, like very side effects also can be, consequences can be expected. So the generalized use of nanomaterials could create a nanopollutants different from those provided by suspended macro or micromolecular particles. So the nanopollutants can be harmful, such as from the cadmium and other heavy metals, or core materials that are used for capping of the nanomaterials could be more toxic to the people. So we should be like, there should be more and more obstacles to overcome the implementing this technology for coming usage, and the science should be constantly refining, developing, and the make breakthroughs. So finally, moving to green nanotechnology, like we'll be focused on nanomaterials as well as methods free from deleterious environmental side effects and potential risk for operators. And we must think on the sustainability of our research effort, focusing on natural and original nanomaterials and soft techniques, which besides reducing the risk and improving the economic opportunities. So as of now, we have a lot of industrial pollution and water pollution, which is leading to our life is becoming very difficult in the coming years. And with the use of those nano-based materials like nanotechnologies, green technologies may become earth very green and sooner. Hope for the best in coming technologies will make the better society for us to live. And so one of the personal responsibilities we should, as a human being, we should think that world is only one for everyone. So we should be very careful whatever we do. We should not making more pollutions and damaging the earth. And we should realize that if we can minimize the amount of damage that we can do the earth, we can live on the earth. And we should reconciling the idea that we might have to change some of our behaviors and habits to make our earth more green in future. And we should until we look for some planet that we can accommodate, we should save the earth until that time. So thank you everyone for your time, and thanks for the organizers for giving us the opportunity to talk on something new that is different from my research. And thank you for everyone.
Thank you, Dr. Rao. If someone has any queries, he or she can.
Yes, ma'am. Myself, H.N. Singh. मेरी कोई सर ये जो वाटर प्यूरिफिकेशन की यूनिट आपने बताया, इंडस्ट्रियल लेवल पे इसको सपोज सम इंडस्ट्रीज गिविंग वेस्टेज, तो क्या उस वहां पे इसको इंस्टॉल कर सकते हैं हम कि जो प्योर वाटर है वो रिवर में जाए?
Yes, like that is one of the system where the flowing water can be purified. Like
right.
We can, yes, that is already
at a relatively large scale, then it will be much beneficial. अच्छा सर सेकंड क्वेरी थी कि जैसे एक इलेक्ट्रिक कार का आपने बताया, तो इलेक्ट्रिक कार में जो बैटरी सिस्टम है इंस्टॉल करने में जो हम उसको बनाएंगे तो उसमें कार्बन इमिशन की बात है, तो एक कंप्लीट पैकेज के लिए कार्बन क्रेडिट का कुछ इवैल्यूएशन हुआ है कि नहीं, कि जैसे एक बैटरी को हम कंपेयर कर सकें कि कन्वेंशनल में उसको रन करने में ये कार्बन क्रेडिट है और बैटरी से करने में ये कार्बन क्रेडिट है, और दूसरा जो इलेक्ट्रिक गार्बेज क्रिएट होगा उसकी रिसाइकलिंग सिस्टम, क्योंकि यदि वो रिसाइकलिंग हमारा नहीं होता है तो अगेन वी आर शिफ्टिंग फ्रॉम वन प्रॉब्लम टू अदर रादर देन वी आर एड्रेसिंग द प्रॉब्लम.
Yes, that is the point of my making, that people are marketing it as a green, but when it comes to the reality under the ground, it is not green really. So
right. We are shifting from one to other. तो इसमें यदि उसका प्रॉपर रिसाइकलिंग सिस्टम हो तो दैट विल बी आवर गोल विल बी अचीव्ड.
Yes, yes.
Thank you so much, sir. Thank you so much.
Thank you, sir.
So I have a quick question. You know,
Yes, sir.
one of your slides you showed like, you know, the electrode and, you know, have the bacteria killing thing.
Yes, sir.
So if you need to scale up, do you foresee any issues with that? Because you may have done, or whosoever has done is a very small scale, but, you know, it's very cool idea actually.
Yes, yes.
So you have to scale up, you know, what do you foresee any issues?
I think so, sir. I think the whatever the material they have developed, like the hybrid material, like nano hybrid material, so that is like found to be very effective at the small scale, whatever they done. But large scale productions can be easy, I guess. Like it's not a bigger issues until unless they made some tube-like structures, like where the water is flowing from until the supplying, like even in the homes can be installed, like from the tap water, like if you are conducting something like, so very small regions like where we have a groundwater is polluted, where they don't have accessibility to drinking a healthy clean water, we can have a separate devices or small units where they can purify those waters and can be accessible to the low economic countries, peoples. Like.
Yes, because, you know, the water, you know, good quality water is so important, because 70% of our body is water. If you don't have good water, you know, then your health is right there.
Yes, sir. Yes, sir.
Okay, good. Thank you.
सर मेरा भी एक क्वेश्चन है कि हर चीज में नैनो टेक्नोलॉजी इन्वॉल्व हो गई है, तो क्या ये कॉस्ट इफेक्टिव टेक्नोलॉजीज हैं?
अभी जो रिसेंटली जो है everything should be like cost effective methods are coming up because of the changes in the methodology and changes in the process development, and also use of more of eco-friendly materials such as plant-based products now, like as I mentioned, plant extracts, and even the microorganisms such as bacteria and fungal organisms. So they also have a catalyzing property where large scale material can be compared to nanoscale material. So it is making more cost effective and more healthier like than the previous one.
एक क्वेरी में से सुनीता मैम से थी हमारी, मैम हम पार्टिसिपेंट्स को सर्टिफिकेट मिलेगी कि नहीं, क्योंकि हम तो पेपर प्रेजेंट नहीं कर रहे हैं.
सर पार्टिसिपेंट्स को पार्टिसिपेशन का मिलेगा और जिन्होंने पेपर प्रेजेंट किया है उनको पेपर प्रेजेंटेशन का मिलेगा.
थैंक यू मैम. थैंक यू मैम.
सर ठीक है सर, उसकी इन्फॉर्मेशन व्हाट्सएप ग्रुप पे दे दी जाएगी, आप लोग परेशान मत होइए. थैंक यू. Now, due to some urgent work, our next speaker, Dr. Ranjana Kanchan, will not joining us for live, but she had sent her presentation. But before starting her presentation, I would like to call upon Dr. Sonal Agarwal for her introduction. Sonal?
Yes. A very good morning to all the participants who are present here with us today on the second day of the International Conference on Chemistry, on Health, Hygiene and Environment. I am Dr. Sonal Agarwal, Assistant Professor of Psychology in Navyuga Mahavidyalaya, and I am delighted and honored to present before you our next eminent speaker of the day, Dr. Ranjana Kumari Kanchan. Dr. Ranjana is presently working as an instructor in the Department of Biochemistry and Molecular Biology in the University of Nebraska Medical Center, Omaha, USA. She has done her graduation from MJP Rohilkhand University and her post-graduation from Hamdard University, Delhi, India. She has completed her PhD from Central Drug Research Institute, Lucknow, in the field of toxicology, and has also been awarded the Junior and Senior Research Fellowship during her pursuit of her PhD. She has gone on to do her postdoctoral from the University of Nebraska Medical Center, USA, and has received several awards and honors, including the Postdoctoral Fellowship Award in India. She has presented her research papers in several reputed national and journals with very high impact factor, as also in various conferences. She did a lot in the of breast cancer brain metastasis targeted therapy projects, and I look forward to sharing to her presentation in our conference today. So, and thank you.
Dr. Sonal. Now I am sharing her presentation. Is it? I think there is some connectivity issue. Sunil ji, are you there? Sunil ji? Sorry for the inconvenience. Yes, sir. सुनील जी आवाज नहीं आ रही है.
सुनील जी आवाज शायद मुझे लगता है कि आ नहीं पा रही है.
I think there is some technical issue.
Sorry for interruption. I think there is some technical issue in that video. So we would not be able to run it with the voice. So I hope if anyone wants to discuss anything, we can do. Otherwise, we'll be going for a lunch break now. And the fourth session will be started at the scheduled time, that is 1:15 p.m. Sunita ji? Yeah. Can we take a break now?
Okay.
There is a problem. Yeah, so it won't be working.
Okay. Vote of thanks.
Yes.
Yes. From the organizing committee, I, Dr. Sunita Singh, would like to thank our speakers and participants for making this session knowledge-based and interactive one. The talk given by Dr. Sanjay Pandey on RNA-based therapies has for exploring possible avenues in this field. The presentation given by Dr. Gola showed the relevance and need of green technologies for achieving sustainable development goals.
Sunita ji, I'm sorry to interrupt. I think Akhilesh ji will share the PowerPoint presentation. Let us try once again.
Okay.
I'm sorry for the inconvenience.
No.
So Akhilesh Kumar, you are requested to kindly share the screen. Thank you so much.
राजेंद्र कुमार जी, आपकी स्क्रीन शेयर हो रही है.
Kindly bear with us. Akhilesh ji is presenting the screen. I hope all goes well this time. Dr. Sunita? I think we are really sorry for the inconvenience caused to all participants. We have been trying, Akhilesh and many other people are trying to share the screen, but because of the large size of the video, we are unable to run it right now. And we are absolutely sorry for that. I would request Dr. Sunita to kindly conclude the session for the lunch break with the vote of thanks. Thank you.
Okay, thank you, ma'am. From the organizing committee, I, Dr. Sunita Singh, would like to thank our speakers and participants for making this session knowledge-based and interactive one. The presentation given by Dr. Sanjay Pandey on RNA-based therapies has surely given an outline for exploring possible avenues in this field. The presentation given by Dr. Gola shows the relevance and need of green technologies for achieving the sustainable development goals. And I hope all the participants who are presented here and do something in this field, especially taking account of the research perspectives, they are surely benefited in this session. So from the organizing team, I would like to thank all of them. The next session will be at 1:15 p.m. Ma'am, the timing will be?
1:15 p.m. we'll meet again.
Okay. And I would like to end the session with a quote that the meetings of two personalities is like the contact of two chemical substances. If there is any reaction, both are transformed. And I am sure the actions conducted in this conference and the coming part, it will bring reactions among the participants and they will definitely help in their transformation, taking the perspective of their research areas. Thank you.
So let's have a break now. We'll meet again at 1:15. Thank you so much.
Thank you.
Yes, Neha. Shall we start? Okay, okay. Namaskar. A very good afternoon to all of you. On behalf of the organizers, Chemistry Department of Navyuga Mahavidyalaya and the entire Navyuga Mahavidyalaya family, I extend a very warm welcome to a distinguished speaker, Dr. Dilip Upreti. I can see he has joined us in time. And our distinguished guest, Dr. Rashmi Srivastava. So we are into the second day of this international conference, and this is fourth session of the conference. The theme of the conference, like International Conference on Chemistry for Health, Hygiene and Environment. This cannot be more relevant than at this juncture of time when the humanity is faced with the most unprecedented challenge. We all know that challenge is the challenge of COVID-19. Health has come forth as a focus issue in governance as well as the research, because we need to focus. We have realized that we need to focus more, we need to gain more in these field of earlier stages when we were groping in dark about the treatment and prevention of this disease. So chemistry can help a lot in both these fields. Similarly, like it has brought about a paradigm shift. Paradigm shift in the sense that earlier we were thinking about pondering upon the issue of sustainable development that is inclusive of protection of environment and ecological balances. But now we have realized the need that we also need to think about sustainable existence. If we exist and the humanity, the well-being of the humanity has become all the more important at juncture. So in the conference on the first day, we had very interesting and very informative deliberations by some eminent scholars, eminent experts. Like there was this, and the most important feature about these discussions or deliberations was that they talked about futuristic strategy to treat some terminal illnesses like colorectal cancer, and talked about prevention of HIV, not only prevention, eradication of HIV/AIDS. So this was the most recent information that we were able to gain from these scholars, these speakers, which we had heard yesterday. Similarly, there was a very interesting presentation about the medicinal properties of indigenous plants. And this presentation, the speaker also emphasized that we should revive the Indian cultural ethos in our day-to-day life of better and healthy lifestyles. So I'm pretty sure that today also we have very distinguished speakers and eminent scientists to speak, and the session is also going to be very informative. To start with the session, we have the D.K. Upreti. He's a speaker. He's a first speaker in the session. So before inviting him, I would like to introduce him to our Dr. Dilip Kumar Upreti is a emeritus scientist, SIR. Earlier, he has worked with NBRI, National Botanical Research Institute in Lucknow. His field of expertise are biosystematics, climate change, bioprospection, and biodeterioration studies. And basically, from reading from his biodata, I can see that he's like passionate about studies of lichens. And he has many landmarks. He has created many landmarks in the field of study of lichens. He is as a distinguished scientist. He has 350 research publications to his credit, and they also include 13 books. He has a long span of 40 years working as scientist, and he is still going strong, and we wish him good health, and he should continue his research work. So during his 40 years of research, he has been awarded with many distinctions, many awards. Like he has been a fellow of National Academy of Sciences in 2018. FNASC award, he has received FNASC award in 2013. And he has been fellow of Ethnobotanical Society in 2010. He has been awarded for his distinguished research by UGC, University Grants Commission, in 2013. He is also a recipient of B.A. Razim Medal Award 2006. He has been president of Indian Lichenological Society. He has conducted 37 research projects in his research career. He has visited many countries like Germany, USA, South Korea, Ukraine, and he has been a member of Antarctica expedition to the South in 2000. In 1992, I believe. Dr. Upreti, the most distinguished achievement of his career that he has discovered 104 varieties. He is pioneer in in the field of Indian study of Antarctica lichens, and he is also pioneer in lichen bioprospection. So these are the fields which he holds expertise in. So today he'll be talking about the use of lichens in pharmaceutical industries and how we can utilize these lichens for deriving drugs for well-being of human beings. So I would invite Dr. Upriti for his deliberation.
Namaskar. Good afternoon. Dr. Sushma, thanks a lot for good words you have used for me. It's really an honor for me, and that your college has given me this opportunity to talk or to tell something about this lower group of plants, which is quite neglected, not known many people, because a lot of work has already been carried out on angiosperms, the flowering plants. We all know, we are more familiar to angiosperms, but these lower group of plants, they are neglected. They are not well worked out from this country. So we use these lichens every day. If you use garam masala, meat masala, yes, it's 90% constituents are lichens. If when you were in high school or intermediate, you have used that litmus paper, that dye was extracted from lichens. It is a dibenzofuran. So we know much about this plant academically, but we use this commonly, but we do not know the uses. So my lecture is on lichen secondary metabolites, how these lichens, they are unique plants and how they produce these secondary metabolites, and how they can be used for development of these drugs, whether through extracellular, we call them lichen acids or lichen substances. So within primary metabolites, yes, we know they are synthesized both by algae and fungi, and they are very quickly utilized by the plant itself in its metabolic activities. Then they are bound in the cell wall or protoplasm. They are water soluble, and they are non-specific, also occur in free-living fungi, algae, and higher plants. Protein, amino acids, we all know, polyols, carotenoids, polysaccharides, and vitamins. So among this, particularly these amino acids, certain lichens, they produce a special type of amino acids. We call them microspring-like amino acids, which are excellent material or chemicals to develop UV creams, because this type of amino acids are only found in lichens, which are growing at very high altitude, up to 4,000 meters on rocks, which are exposed to the high UV. Next, secondary metabolites, they are fungi. There are three types of fungi involved in lichenization. Ascomycete is the base type, and about 50 of see 500 secondary.
Dr. Upriti, there's some issue with your microphone.
Voice.
So your voice is not clear, sir. आवाज सही से नहीं आ रही.
Okay.
Yeah. So I think we are not able to hear you. Your voice is not clear. There's some issue with the microphone, probably. Attendees, please bear with us. These type of technical glitches happen often, and we are sorry for that.
Is audible? It
No,
is
sir,
audible?
not yet. Yeah, please, sir, please you continue, then we'll be able to tell you.
So significant.
No, sir, your voice is disconnecting.
सर मे बी नेटवर्क इशू है थोड़ा, एक बार फिर से कनेक्ट करके देखिए.
सर मे बी रीजॉइन करने से कनेक्ट हो जाएगा. So your slide is visible, but we cannot hear you.
सर अनम्यूट कर लीजिए.
Maybe your mic is muted. I hope sir will be able to join us soon. Please bear with us.
Presentation mode. Stop sharing. I'm audible?
Very much, sir. Very much. Welcome back.
Okay. Slides are there?
Yeah. No, slide is, yes, slide is also visible. No,
Okay,
slide is
okay,
not
I was
visible,
talking-
sir.
It's
So we
visible
cannot
or
see
not?
your slides. No. We cannot see your slide, but we can hear you. Sunita, I think slides are not visible.
स्लाइड्स से देखिए सर, वो आप टैब में खोल के रखिए तब आप कर पाएंगे, या तो इंटायर स्क्रीन ऑप्शन से करिएगा. इससे पहले दिख रही थी स्क्रीन, आपकी आवाज नहीं आ रही थी.
Slides are visible?
Let us see, sir.
Slides are visible?
No,
Sir, not yet. Yes, sir.
Yes,
Yes, sir.
Visible. Yeah. We can slides.
Okay. Okay, I was talking about the significance of secondary metabolites. As I told you, lichens, there are 20,000 species of lichens so far known from the whole world. And if you see these 20,000 species, they produce more than 1,500 secondary metabolites, out of which 100 or 50 are common with other group of plants. Otherwise, 1,350 are unique. They are not produced by any other group of plants or other organisms. I'm audible and slides are visible?
Yes, sir.
I'm audible. It's a clear voice.
Yes, sir, you are audible. Sir.
Okay. Okay. We can use these secondary metabolites when we go for taxonomical studies. Sometimes certain, if you go to the systematics or taxonomic studies of lower group of plants, in case of higher group of plants, you have too many characters for segregation, whether because higher plants, they have leaves, they have flowers. So depending on different type of flowers, different type of leaf arrangements, we can separate different groups. But in case of lower group of plants or up to the bacteria, we do not have many morphological characters. So similarly in lichens, we do not have too many morphological characters. So chemistry plays a very important role over there. So we use them in chemotaxonomy when we go for different stains of particular plants, or even biochemical systematics, we use the chemistry of the secondary metabolites. Then we use the use of lichens in medicine, in dye. They are very useful because for herbal dyes, the lichen chemicals have an ability that they can color a protein or a fiber which is animal originated. And we all know about its value of perfume. In Kannauj, a large number of these lichens, they are exploited for Hina Atar, a perfume which is very highly priced perfume from India. So it is, we all know that it is the lichen which produces chemicals, secondary metabolites or primary metabolites. The role which is played by algae is more important over there. So if you go to the 5 synthetic pathways which were discovered earlier, it was Mosbach who summarized carbon metabolism in lichens. Then it is William Culberson of USA. He provided a systematic presentation of the biosynthetic pathway of secondary metabolites. Blue-green alga, it supplies glucose. If that lichen comprises of cyanobacteria or a blue-green alga, it will produce glucose, while green alga supplies polyols, rhabitols, sorbitols, erythrols, and mannitols. So from this picture, it is clear. We know that alga is there. By the photosynthetic process, it manufactures food for fungi. That is, it may be glucose, erythrol, rhabitol, or sorbitol. Then it passes to the fungi. So depending on the type of fungi, there are 3 major pathways are involved. Acetate polyvinate pathway, malabinic acid pathway, and cyclamic acid pathway. You can see that the compounds, dipsidezones, dibenzofurones, they are not produced by any other group of plants or even the other organisms. So what happens if you see this is major classes of secondary metabolites as polymalone pathway. Metabolites, derived aromatic compounds are there. Malabinic acid pathway, yes, diesters and esters are there. Steroids, they are produced from this pathway. Then cyclamic acid pathway, it is particularly the polyvinic acid or yellow pigments. So acetyl polyamine pathways include most of the ligand substances formed by bonding of 2 or 3 organol or beta organol types. Then I was talking about these dipsidezones, dibenzofuron, arsenic acid, dipsones are peculiar to lichens. They are not produced by any other organism. Then chromones or xanthones or anthraquinones are there. They are produced by other group of organisms too, by higher plants. We all know about that these anthraquinones, quinones or chromosomes, they are produced by higher group of plants too. So you can see the different structures of acetyl polyamine pathway compounds, that is protolycrastic acid, oxalic acid. This oxalic acid, they are mostly used for dyeing this ability. The lichen which comprises of this oxalin or oxalic acid, they are used for dyeing. Malabinic acid pathway is a common product include steroids and triterpenoids. Geurine is most common. We encounter mostly this geurine triterpene in lichens. And again, the lichens which has this giorin triterpene, they are considered as toxic tolerant to the pollution. Means in polluted areas, lichens do not grow. But if they contains this giorin or triterpene, lichens have an ability that they can withstand that environment which has some pollutants, and lichens able to grow over there. Sequemic acid pathway, this pulvinic acid derivatives, they are most common yellow pigments. And the lichens which are yellow in color, they are peculiar of this pulvinic acid. So how we identify these metaboloids? There are two methods too. Means we use this spot test. In the spot test, we have potassium hydroxide 5%, then diamine. When we put
sir, it is stuck on significance of secondary metabolites.
it's moving.
So
You just need to go
moving.
to next slide. No, sir, not yet.
There is some
Slide
light
number
problem.
10 is visible only. You just need to go click the next slide. And
Yes,
it will
sir.
appear.
It's visible. Next one.
No, sir.
Not yet, sir.
Let me see. We can do this, yeah? No. So, you just need to click slide number 11.
Oh, we are clicking it. Yeah, slide number 11.
No, it has moved way too forward. Now we have come to slide number 18.
18. Yes, sir.
Yes. Next.
Hmm.
Slide number. It is identification of secondary metabolites.
Yes.
Yes, sir.
So I was talking about that how we can identify the lichen compounds. There are some easy methods, particularly one spot test we call it. What we do, we use 10% of this potassium hydroxide solution, and we called it K. Then we have paraffin diamine. When we put K, the dipside and dipside regions, if they are present on lichens, they give a color. They give a yellow or deep red color. And other reagent we use, it is calcium hypochlorite, means bleaching powder. When we put this bleaching powder on certain lichens, if it contains some phenolic compounds, it gets a red color. So this is a very simple method to identify lichens. Even like compounds are very easily develop some crystals. So we use to study these crystals, and based on these crystals, we can identify. Then again, we have all these methods, paper chromatography, TLC, these are other methods by which we can identify lichen secondary metabolites. I'm audible and slides are there?
Yes, sir, very much.
Okay, next one.
Well, yes.
Yeah, this is lichen species. And what are the lichen species? One drop on that lichen, it immediately color. Then calicine, pulvinic acid, they are most common compounds, or I have given the list of most common lichen chemicals and lichens which are there. If you see the statics of lichen, medicinally important lichens in world, we have only 500 lichens on which, whether it is the medicinal information is available through pharmacopoeias, or medicinal information on those lichens are available from ethnic groups, folklore, or even what we have carried out recently, the preliminary studies on medicinally important lichens. So out of these 500, we have about 160 species of lichens which are medicinally important in India. It is quite meager. If you see the number of lichen species which are growing in India, India is considered as a mega diversity country, and apart from other group of plants, lichens also exhibit much diversity or rich diversity in our country. So out of these 3,000 species of lichens, only information about 160 species of lichens are known which are medicinally important. Is it visible? Slides are visible? Lichens and the doctrine?
So we are at the identification of secondary metabolites. We need to move.
उसी से उधर से चल यार वहां से जो साइड नेक्स्ट it is there now changing
Not yet, sir. It is slide
Changing?
number 18.
यहाँ से कर
So please click slide number 19. Yeah, it's visible, sir. It's visible.
So I was talking about the lichens and their common lichens and their chemicals. So mostly we can, on the basis of their color, we can group them into 3 or 4 groups. Means lichens which produce dipsides, lichens which produce phenolic compounds, and the lichens which are yellowish in color. Is changing? Slide changed or not?
Yes, it has changed, sir. It has changed.
Next. So I was talking about the status of medicinally important lichens in India. And if you see that out of 3,000, I was talking about that out of 3,000 species of lichens known from this country, we have only information on 160 medicinally important lichens from this country. Next, it is doctrine of signature. Means we can even get leads from certain informations, what we call doctrine of signature. The nature has given certain plants or made certain plants which are similar to the human organs, and they are used for treatment of that particular organ in the humans. For example, one species, one lichen is Peltigera canina, means its fruiting bodies are similar to the canines of a dog. So this particular lichen is used in treatment of rabies. Lobelia pulmonaria means the structure of that lichen, Lobelia pulmonaria, resembles our lungs. So this Lobelia pulmonaria lichen is used for treatment of some lung diseases. So we call this doctrine of signature, and we take it from such informations to evaluate the medicinal properties of different organs. Slide changed. Slide number 25 is there.
No, sir, it's 22.
Hello.
Yeah, now it's 23. Slide
Yes,
number 23.
Lichens in ethnomedicine. Yeah, I was talking about that. India is very rich in ethnic, in different ethnic type peoples. We have more than 500 ethnic groups in our country, and they have extant traditional knowledge. And they have knowledge on these lichens too. So we get leads from those ethnic informations or from folklores where the lichens are used for treating skin diseases, even bone fracture, insecticide in kidney stones and rabies. So we have mentioned and we have listed that more than 60 lichens, they are frequently used by different ethnic groups in our country. And more than 40 different diseases can be cured with lichens. So it may be an antiseptic, it can be used an anti-diabetic, antifungal, cold and cough, anti-asthmatic. So we have more than 40 diseases which can be cured with lichen compounds. So the most common lichen, if you go to higher Himalayas, you will find some lichens, particularly this Cladonia furcata. And in tundra region, it grows like grass, heaths, and the reindeers, they feed on it. So in our country also, this Cladonia furcata is there, and it has anti-cancerous and cytotoxic activities they are reported. Then Cladonia rangiferina, yes, again, it has certain chemicals. That chemical is particularly one fumoprotocitric acid, which is used in treatment of kidney stones. Then this particular species is very common. It is very frequently used in spices in our country. We call this is sold in market as chadila. And again, it is used in a large number of treatment of different diseases by ethnic groups. Parmule falcaprata, again, it contains certain chemicals which are used in treatment of dementia or even arthritis. So the chemical present, and we are working on these aspects too. Then heterodermia diademata, this contains a georine, a triterpene, and that particular lichen is used for treatment of skin diseases, and which are mostly related with the fungi. Antifungal activities are well known from this lichen. I was talking about Lobelia pulmonaria. You can see that this is different species of Lobelia, but has a similar structure to our lungs. So it is again very frequently used in treatment of different lung diseases, and Chinese, they use it. Then Parmotrema perlatum, if you see the old literature, even if you see the calendar in Himalayan drugs, you will find that they have used this lichen for treatment of different diseases, and but this is not very commonly growing in India. But the next one, if you see, I was talking about this Parmotrema reticulatum, which is used in Hyderabadi biryani for flavoring here biryani. So this is very frequently used as spices. Then Peltigera canina, I was talking about that this particular lichen, it has canine-like of fruiting bodies and used in rabies. Again, Ramalina conduplicans, it is again very commonly growing lichen in our country and have a very potent antifungal activity. Rossella montagni, if you go to the main groups, you will find plenty of this lichen, and it has very then comes the Stricta nylandriana. It has certain these phenolic compounds which are used as in asthmatic in controlling cough and asthma, and you can see that frequently this name is appeared when you use the syrups from Badnath or from these other local Indian medicinal syrups. Thamnolia vermicularis, yeah, it appears like a worm, and again it has certain vermicidal activities are there. Usnea longissima is the most frequently used lichen for its medicinal properties because it's contains usnic acid, which is very useful. And again, there is a drug is available from this plant. Then Xanthoria peridina, if you see, is a yellow colored and very commonly growing in Ladakh. Here, the Doctrine of Signature, because due to its yellow color, it is used in treatment of jaundice. Now, depending on this information which we have, now we go to the evaluation of these drugs, and we have taken out one quercetin. It is a flavonoid from a lichen Usnea longissima, and we developed a nanomaterial for treatment of Candida albicans. Candida albicans is a skin disease which cannot be cured by the fluconazole ointment which is available in market. But when we have developed the nanoparticles from lichen quercetin and mixed it with fluconazole nanomaterials, it can treat Candida albicans in its all growth forms, whether it is saccharomyceal, it is myceal, or it is virulent, because the fluconazole which is available in market can only control mycelial type of infection on the skin disease. But nanomaterials which we developed from these lichens, and again I will tell you, lichen chemicals are very suitable for development of nanomaterials because they have too much of functional sites, and which we can easily develop a nanomaterial. So we have worked on this particular aspects, that how these nanomaterials can be developed from lichens and taking their chemicals and for treatment of different skin diseases. Even we know about Swarnavasma. This Swarnavasma can be developed from lichen compounds. Lichens, particularly again, we had used Usnea longissima, and we have extracted or we have developed certain nanomaterials, gold nanomaterials. We know that earlier this Usnea longissima dust was used for treatment of dental caries. We all know Staphylococcus mutans is responsible for that, and we have, when we develop this herbo material Swarnavasma from lichens, it can reduce or it can control these dental caries in humans. There is a term about nanofactories. Again, the lichen compounds, natural compounds, and we have used here fumer protosyntenic acid because now due to the superbug, a single nanomaterial cannot kill that superbug. So there is a need, we can go for assemblage or aggregation of 2-3 these nanomaterials. We have taken from honey, we have taken from curcumin, then we have taken one natural product that is fumer protosyntenic acid from lichens, and we develop this nano scaffold which can destroy Pseudomonas, a deadly pathogen in its initial stage when there is a biofilm formation is there. So we have worked on hepatoprotective activity of lichens too, means a lichen which is Cladonia furcata, which is growing very frequently, can be used in treatment of in gastroesophageal reflux diseases. So we have worked on these aspects too. Next. Now, as I told you that lichen is not a symbiotic association between algae and fungi. There are a large number of other fungi are too which are living inside lichens. If one lichen thalli comprises of these fungi, we cannot see any symptoms outside. But these fungi, they are living inside, and they are excellent source of a large number of these alkaloids, polyclols, or steroids. But there is a need of synergy that one taxonomist, one biochemist, or a phytochemist can sit together. Lichen, about lichen, one taxonomist will tell, but the chemist will tell about the different chemicals. Then trace out the pathways which are responsible for production of these chemicals. Then we need a biotechnologist who can transform that particular gene to a microbe to get good biomass of that particular compounds. So lichens, and particularly the fungi which are living hidden inside the lichen thalli, are excellent source of pharmaceuticals in future. Next. You can see that some pictures of these lichen secretory metabolites, and you can see that there are too many, these functional sites are there, or these polar groups are there, OH or COH, or they are excellent source to develop nanomaterials, as I told you earlier. In India, we have large exploitation of lichens, and more than 10,000 tons of lichens are collected every year and destroyed. Lichens are very slow growing. So for conservational point of view, we started culturing lichens in laboratory conditions, and we are successful, and we got the same, in most of the cases, the same secretory metabolite which is present in natural thalli. So that can be used for different bioprocessor or for commercial uses. So we can grow them in petri dishes. We can grow good biomass in broth as broth culture. And if you see that, so depending on these studies, you can see that a large number of lichen products are available in market. As I told you, lichen spices available in Indian market, garam masala, meat masala. Then we have edible and medicinal lichen products, which are, I told you about Asno. We have the lichen blend products. There are a number of perfumes. Then Asnia, which is used for development of one ointment for skin disease, Asno. So this is all about lichens and their pharmaceutical potential. Sorry for intervention. There is a problem in this technical problem here. Anyway, I have finished my lecture. Any questions, any clarifications are welcome.
Thank you very much, Dr. Rupreti. It has been a very enlightening, very informative lecture by you, especially for the lay people who are not who do not know about the properties and the use of lichens. So now I open the floor for question and answers. I would like you to directly address your question to our eminent speaker. If there are any questions from attendees.
Sir, may I ask a question? I just want to know, sir, like you told about the nanoparticles from lichens. I mean, the nanoparticles which are developed from lichens, they have they treat wonderfully those diseases which piconazole, like other ointments, are not able to treat, which are available in the market. So, sir, I just wanted to know whether do we have any side effects of these nanoparticles or or not?
Yeah, yeah. I will tell you what we have done. We have done with fluconazole. If you see this fluconazole ointment, which is available in market, is used for this outer means on it. We cannot get inside, but we can use for treating skin diseases and all. So it is well known that certain heavy metals, certain nanoparticles are not used, or the drugs are not used directly for taking inside our body, but for skin diseases, and for that particular purpose we can go for that.
So one more question I have. There are some recurring infections in the skin, like fungal infection and all. So do we have any solution for that from lichens?
Yes, we have information from the ethnic groups that they use frequently lichens for particularly ringworm type of diseases, and since their availability is not the biomass is not great in nature, so it is difficulty is to getting these by these ethnic groups now, because earlier they were frequently used. Now, as I told you, that lichens are very slow growing. A lichen assume a size of 1 millimeter in 1 year sometimes. And certain lichens are there which are growing in the summit of the rocks in near glaciers. They assume a diameter of a size of 1 millimeter in 100 years. That property we use in climate change and other studies, but the growth is very slow. That's why we are going for these nanomaterials and all. So these informations or these leads are based from the ethnic groups also. Okay, so this particularly the fluconazole, which is available in market, can cure when the Candida albicans disease is mycelial. But if it is virulent, if it is saccharomyceal, it cannot be controlled with this fluconazole, which is available in market. So what happens? The quercetin or flavonoid we extract from Usnea longissima, because Usnea is well known for its antifungal activity on skin diseases. So the flavonoid which we extracted from lichens, we characterize it, we isolate it, characterize, then we develop the nanoparticles both from this fluconazole and Usnea, this quercetin. This quercetin has increased the production of furcinol, which is a compound present in fluconazole to kill all the three growth forms of that fungi. So it is there. That is particularly the kinetics which is involved in this particular material.
So, any more questions? I think there are no more questions. So I would like to formally thank Dr. Upreti. You presented a very exhaustive and comprehensive account of lichens, and you talked about the variety of lichens, their provenance, where they're grown, and especially the interesting part was that you told us about the day-to-day use of lichens. Besides the pharmaceutical importance, you talked about the day-to-day where we are not even aware that Hyderabadi biryani owes its popularity to lichens, and similarly most of the herbal products which are very much in vogue in cosmetics, they also have lichens as their components, like most of the skin and skin products and all. So you discussed that we have around 3,000 lichens, only 160 lichens have medicinal properties. So it is always better to go for herbal. So I think more researchers in lichen studies will enable us to revive our traditional knowledge and to produce safe medicines, because we know that most of the medicines claim modern medicines claiming quick relief and quick cure, they are basically using some very harmful chemicals. So it would be always better to switch to natural products, and I think lichens are very good alternative for us. And you also told us that this is not something new to Indian knowledge system, because ancient medicinal tests, they also talk about shell and shell push, etc. They're actually lichens. You also told us about, you emphasized that we should depend, or we should try to harness the ethnic communities, the knowledge of ethnic communities about the lichens, so that that can be brought to the mainstream of pharmaceutical field, and they can be harnessed and they can be utilized in a proper way. So, Dr. Preeti, it was a very enlightening lecture, very knowledge imparting, and we are all so very thankful to you. We are especially thankful that you are such a busy person, and still you accepted our invitation, and you gave your valuable time for us. Thank you very much for being with us. Namaskar.
Yes, thank you. Namaskar.
So now our next segment of this session will be oral paper presentations. And for this section, I invite a very well-known and very again an eminent scientist, Dr. Rashmi Srivastava. And Dr. Rashmi Srivastava is basically a paleobotanist by profession. She did her PhD from Lucknow University in the field of botany. Later, she specialized in paleobotany. She also has a very distinguished career as a scientist. She retired as a scientist F from Birbal Sahni Institute of Paleobotany. She has worked on her field of expertise is angiospermic fossil flora that comes from 60 to 67 million year old rocks from sequence of Madhya Pradesh. She has been a very active scientist. She has published around 85 papers and attended 50 scientific research conferences and symposiums. She has many awards to her credit. She has been awarded ENSA JSPS International Scientific Fellowship in 1998 and ENSA Polish International Scientific Exchange Fellowship in 2010 to visit Japan and Poland. She has been very active in Indo-US collaboration program, which is under the National Science Foundation of USA, and she has visited many countries. Besides working in many parts of India like Himachal Pradesh, Kerala, Gujarat, etc., she has visited many countries abroad for her purpose of research. I know how difficult a career is for a woman in paleobotany, because traveling to remote areas, negotiating rugged terrains, and working shoulder by shoulder with men in the thick and thin of the weather, I can understand. So you are the kind of women who are breaking glass ceilings and creating an ideal for young scientists and young women, that nothing is physically impossible for women. They can do whatever men can do, and maybe they can do it in a better way. So I welcome Dr. Rashmi Srivastava. And now we will be starting a session of oral and poster presentations. For this, I think I should invite Dr. Vimla Ben to introduce speakers.
Ma'am, I just want to interrupt here. Actually, Vimla Ben is not available right now. So I would be taking the session for oral presentations.
All right, it's okay. You're welcome, Neha.
Thank you, ma'am.
Rashmi, your mic is not on. Please unmute your mic.
Thank you, Sushma ji, for your kind words. And first we can presentation.
Yeah.
Okay.
So the first presentation is oral presentation, Ashutosh Mishra.
Okay. First thing I would like to tell you to all the participants that because of time constraint, please restrict your presentation to 3 to 4 minutes. And if time will exceed, negative marking will be done. So please take care of this. Thank you.
We should stick to time because we have a very exciting upcoming valedictory session. So we all need to stick to our time. Yeah.
Ashutosh Mishra, are you there? The next participant is Dr. Pooja Srivastava. Dr. Pooja Srivastava, are you there? The third participant.
Hello.
Is
Yes, ma'am. I'm here.
Please start your presentation.
Ma'am, hello.
Yes, please.
Ma'am, is it possible to present after half an hour or so? Just there.
Since it was already informed that we'll be doing according to the sequence which we have already given. So it
Yeah,
would be actually
ma'am, it
difficult for
was-
us to reshuffle the numbers. So I would request you kindly present right now.
Okay, ma'am. Just give me 2 minutes. I'm sharing the screen.
Hello, Nehaji. I'm unable to see the slides.
Yeah. Pooja ji, will you please share your
Yeah,
screen?
ma'am, I'm sharing. Is it visible, ma'am?
No, not yet.
No.
The slides are not visible. My screen is visible or not?
Yes.
Are
Yes. Now
visible
it is
to us.
visible. Now it is visible.
Okay. Actually, internet was not working here, so there was some problem. Some people were trying to make it work. So that's why I asked for 15 minute or 20-minute break. Anyway, so I'm starting. I hope that it will work. My phone's hotspot is working. So I hope it will work throughout the presentation. Okay, so good afternoon, everyone. And today I will be talking about the two-dimensional materials and how they can control the air pollution. So mainly my presentation will be about how they can control the air pollution. And since I am a theoretician, I do DFT calculations. So my study will be mainly focused on the theoretical results, which I myself have calculated. So enormous emission of the carbon dioxide due to excessive consumption of carbon-rich fossil fuels cause pervasive and irreversible environmental changes, and its primary reason behind the dangerous fluctuations in global temperature and climate. An attractive solution to this problem is to convert CO2 into useful chemical. For example, formic acid, it is very useful chemical and we all know about it. And also the fuels like methane or methanol. Now, let me come to the nanomaterials. So here I will be talking mainly about the two-dimensional materials, which are a kind of materials. You can say that these are the upcoming nanomaterials where the in-plane interaction is very, very strong compared to the out-of-plane interaction. So because of this reason, they are called two-dimensional material, and also they are very thin also. But there is a difference between the surface or this layer and these two-dimensional material. So the examples include graphene, MOS2 or MaxScenes. And one thing I want to tell you, all of you, that the two-dimensional material is not a single material, but it is a family of material, and actually they cover the full range of spectrum. So in the 2D materials family, we have metal, we have semiconductor, we have insulators. So because of this reason, they can be a potential candidate for various kind of functionalities like catalysis, sensing, or they can act as support for nanomaterials. They can be very good for electronic also, optoelectronics. So there are many areas where they can function in very good way. In the present work, I shall be focusing mainly about the catalytic potentials of one particular kind of MaxScene family. Since time provided is very less, so I have chosen only one kind of two-dimensional material, which I am calling MaxScene. So it's also a family. I will discuss more about the MaxScene in a minute or so. So these are the two-dimensional layers of early transition metal carbides or nitrides. And these materials are very good for carbon reduction reaction. So they can catalyze this reaction. What is good about the Maxine? So they have the combination of properties of ceramic and metal. So being a ceramic material, it is highly stable material, and being a metal, it is highly conducting material also. So as I said, that Maxine family, it is a kind of family, not a single material. So Maxine family is gaining immense research interest, not only for catalysis, but for various functionalities like as I mentioned earlier also, they can be a good optoelectronics material or electronic material. So these materials are used for the catalysis of many different kind of reactions. CRR I have already mentioned, HER hydrogen evolution reaction, water splitting, for many reactions they are working in good way. Recently, the formation of formic acid from CO2 is observed in the when the CO2 is passed over the Maxine. So it is reported in an experimental study. In fact, there are many theoretical studies which are already predicting the conversion of CO2 to methane, methanol, formic acid. So now let me come to Maxine, because I'm talking about the Maxine, but I have not clarified that what is Maxine. So Maxine is basically, as I said in the beginning, that it is a kind of 2D material, and it is synthesized from the Max phase. So here you can see the Max phase. So M stands for early transition.
Excuse me, you just finish your lecture because time is already running off.
Yeah. Okay, ma'am. So Maxine is prepared, synthesized from the Max phase. So as I said, M stands for the early transition metal, A is aluminium, and X can be C or N. And the most importantly, it is not the van der Waals solid. So it is difficult to find the Maxine. Here we have to etch away the A layer. A layer means Al atoms have to be removed from here. So this can be done using the HF solution or LiF plus HL solution. So if you etch it away, this Al layer, then you will get physically separated two-dimensional Maxine layers. So here you can see that except this A, everything is present here. And it is important to know that Maxines are always functionalized. So you will find Maxine only in this kind of form, and usually the functional groups are F, O, or OH functional groups. So here this is the Maxine, and now, as I said, so I will skip this slide. Now let me directly come to the CRR catalysis. Where Maxine works as a good material. So here I have shown a few reaction paths for the formation of CH4 using the Maxine. So this I wanted to emphasize on this page that, as I was saying, that Maxine is a family of material, and the basic formula for Maxine is this formula. So here the flexibility provided by you can choose M according to your need. You can
Excuse
choose
me, your
X
time is up.
just okay.
Kindly conclude. Kindly
Please,
conclude.
kindly conclude.
So
please, organizers, please, you also
okay,
see
ma'am. So just one thing I wanted to emphasize here. This is my own result. So the main benefit of Maxine is that the Maxine is having a lot of ample number of states at the Fermi level, and also here in this figure you can see that the CO this LUMO of CO is at higher position than the Fermi level of Maxine. So because of this reason, the electron transfer from CO2 to Maxine is very easy, and because of this reason, the Maxine is very good for the CRR purpose. Okay, so I'm concluding, and I'm ending now. So thank you so much.
Thank you, Poojaji. Thank you. Next, please.
Next, Rajendra Kumar. Dr. Rajendra Kumar. Dr. Rajendra Kumar.
I don't think he's present over here.
शायद प्रेजेंट कर रहे थे लेकिन अभी शायद चले गए. शेयर किया था उन्होंने स्क्रीन.
Okay, there are only 2 arts. Rajendra Kumar is not here.
Okay, ma'am. Shall I call next participant, ma'am?
Neha. I think you should call because we need to save time.
Yes, please.
Yes, ma'am.
Yeah.
Yes, ma'am. Neha Chakravarti.
Is my screen visible, ma'am?
Yes.
Yes, now.
Am I audible, ma'am?
Yes.
Yes.
Thank you, ma'am. So first of all, I would like to extend my warm greetings to all of you. Good afternoon, everyone. My name is Neha Chakraborty, and I am a PhD candidate under Dr. Ashna Talwar, Department of Chemistry, Isabella Thoburn College, Lucknow. And I am all here for the poster presentation entitled MIP a Smart Extraction Tool. Before I begin with my presentation, I would just like to give you a brief background. So basically, the natural products and the analogs have made a magnanimous contribution in pharmacotherapy, especially for cancer and infectious diseases. As we all know, penicillin is not unknown to us. According to Mohanty et al., nearly 25% of the major pharmaceutical compounds and their derivatives available today have been obtained from natural resources. But despite it, pharmaceutical companies are hesitant to invest more in natural-based product, drug discovery, and delivery systems. Why? The answer to this why lies in 3 problems related to natural product research, which are isolation, second is the availability of sources, and third is the pharmacodynamics. So one of the key problems of extraction of natural products formulates one of the research objectives of mine, which is to design and synthesize smart extraction tools. Going by the existing methods of extraction, we have Soxhlet method, we have liquid-liquid extraction, and we have n number of micro extraction techniques. But then they all suffer from their own array of limitations. For example, they are very time consuming, they require large volume of solvent, they require large sample size, they have poor recovery, they have selectivity issues, they turn very costly, etc. So one solution that I see to these problems is MIP. So basically, MIP stands for molecularly imprinted polymers. So molecularly imprinted polymers are basically synthesized polymers which have predetermined affinity towards particular class of analytes or a single analyte. So this figure 1 enlists basically the basic steps involved in the synthesis of MIP. So basically, we have a template that is our natural product, and around which the polymerization takes place. Following which we synthesize a template without our analyte, followed by the characterization. So where is the lacuna lying? But still there is a lacuna. But where is it lying? Because the technology transfer of MIP to industry is still zero. So the key challenges that I see in MIP and their potent solution that I feel are, so problem being optimization issues. For example, we have polymer, then we ought to know the monomer. Then out of 1,000 compounds, which will be the suitable monomer? We have to do n number of experiments to know that. This is one optimization issue. So we have what is the solution? We can do multivariate analysis. Second, we have n number of modeling, computer modeling, simulation modeling softwares that we can use. Second, that challenge is the difficulty in access to template. Sometimes the MIP, the template cannot, the analyte cannot access the template because it is too deep. So the solution can be core cell polymerization or the surface imprinting. In our lab, we are targeting steroidal glycoalkaloids of family Solanaceae, which have been reported to have anti-cancerous, anti-inflammatory, antimicrobial, and anti-cholesterol properties. We aim to isolate this functionally, structurally, and chemically versatile class of compound by synthesizing reusable analyte-specific nanotemplates without compromising with the principle of green chemistry. One thing which I forget to mention is that the MIP basically follows the principle of lock and key. We have one lock, we have one key, and the suitable keys fit inside the suitable lock. This is
please sum up your conclusion.
And as I've already told you that we are targeting steroidal glycoalkaloids, and I'm hopeful that within 6 months we will be having some exciting results. Thank you.
Okay, thank you.
Thank you, Neha. Next.
Thank you, ma'am.
Next, Dr. Abhapar. I think she's not there. Okay, Ayushi Pandey, OP05. Ayushi Pandey hai?
Yes, ma'am.
Okay. Please share your screen. And it's a request, please time limit का ध्यान रखिए आप लोग, interrupt ना करना पड़े.
Good afternoon, everyone. First of all, thank you for giving me this opportunity. I am Ayushi Pandey from MIT University, Department of Chemistry, MIT School of Applied Sciences. My presentation topic is synthesis of biologically active heterocyclic pharmaceutical compounds using bioenzyme. Introduction. The use of enzymes in the organic synthesis, especially to make for pharmaceutical compounds as well for the industrial uses. In the most prominent example, biocatalysts are used on a large scale to make the specialty and even bulk of chemicals. Enzymes catalysis is a very energetic research area in the organic chemistry because biocatalysts are suitable with can be adjusted to many reaction conditions as well as a substrate. Their integration many reactions allows for a simple protocol to be implemented in the diversity-oriented synthesis of a complex molecules. The application of a biocatalyst development and emerging as a unique tool in the drug development and discovery related areas of the research, which is environment friendly too. Chemical reactions need an initial input of energy is equal to the activation energy. During this part of reaction, the molecule are said to be in a transition state. This is a reaction in the bioenzymes take place in this first activation energy. In the non-enzymatic reaction, activation energy is more, and in enzymatic reactions, activation energy lowers down, and product are formed and enzyme released from this. These perform the same as the endothermic and exothermic reactions. Enzyme work on the just like an enzyme or protein, act as a biocatalysis. Accelerate the biochem reactions. In this substrate and enzyme, yes, compounds are formed, and at last product is formed and enzyme. The structure in this the active side, the shape and chemical environment inside the active permit the chemical reaction to proceed more easily. Cofactors. Cofactors are an additional non-protein molecule needed by some enzymes to help the reaction. Tightly bound cofactors are called prosthetic groups. Cofactors that are bound and released easily are called coenzymes. Enzymes are specific to their substrate. The specificity is determined by the active site. They are work on the lock and key hypothesis. In this substrate and enzyme behave like a key and lock. Lock and key complexes form like a enzyme substrate complex form, and a product are formed. Fit between the substrate and the active site of the enzyme is exact like key fits into the lock. Very precisely, enzyme substrate complex are formed. Product have a different shape from the substrate. Products are released from the active site, leaving it free from another substrate molecule. The lock and key hypothesis explain the enzyme specificity. And other is a induced fit hypothesis. In this active site, an enzyme conformational change enzyme, then the substrate ES complex and form and product are released and enzyme release. Some proteins can change their shape conformations. Substrate enzymes induces and change the enzyme conformation. Active site molded to precise conformation. The chemical environment is now suitable for the reaction. Importance of bioenzyme: increasing the temperature makes the molecule move faster. Biological systems are very sensitive to temperature change. Enzyme can increase the rate of reaction without the need to increase the temperature. They do this by lowering the activation energy. They create a new reaction pathway, a shortcut, an enzyme controlled pathway. Enzyme controlled reaction proceed 10 to the power 8 to 10 to the power 11 times faster than the corresponding non-enzymatic reactions. Application: enzyme is a chemical and pharmaceutical process. Application of enzymatic process in this chemical industry depend mainly on the cost competitiveness with existing and well-established chemical methods. Lower energy demand, increased product and the increased catalytic efficiency, less catalyst waste and byproduct, as well as lower the volume of wastewater streams in the enzymatic reactions are the main advantage that biotechnological processes have compared to the well-established chemical processes. Enzymes are useful for preparing beta-lactam antibiotics and semi-synthetic penicillin and cephalosporins application of enzymes in several industries. Please sum up your...
Kindly conclude it.
Okay, ma'am. Concluded that the bioenzyme have the potential to efficiently detoxify and transform the pollutant and check the chemical chaos. Despite these favorable condition of bioenzyme, further research is required to bring the innovative idea into reality. The introduction of enzyme as effective catalyst working under the mild condition result in a significant saving in resources and such energy, water, and time for the benefit for the both industry and environment. Thank you, ma'am.
Thank you,
Ayushi.
Thank you, ma'am.
Next, Alka Pandey, OP06.
Yes, ma'am.
Alka Pandey है?
Yes, ma'am. Yes, ma'am.
मैम आप जितने भी पार्टिसिपेंट्स हैं उनसे रिक्वेस्टेड 3 से 4 मिनट ही लीजिए और कंक्लूजन पे आइए कि आप क्या बताना चाहते हैं.
You're not audible. You are not audible yet. Are you sure you're not audible?
Alka Pandey.
Sorry, Alka Pandey is not audible.
आप अनम्यूट कीजिए, आप शायद म्यूटेड हैं. वो शायद आयुषी पांडे के ही प्लेटफॉर्म से शेयर कर रही हैं.
Not. Please.
I think I will call next participant. अलका जी आप अपने एंड पे देख लीजिए, शायद कुछ दिक्कत आ रही है. मैं डॉक्टर अंजुला कुमारी. ओके. डॉक्टर बीबी इशरत जहान, OP08. I think so. One second, Kanchan Singh. Ma'am, शायद डॉक्टर बीबी जहान.
Yes, I am present. Am I audible?
Yes.
Please share your screen.
Yes.
Please start. Your slides are not here. We are unable to see your slides.
Just a minute. I'm trying.
think it is not open.
You have not started your presentation?
My PPT is not opening. I don't know why.
Okay, I will call next participant, Dr. Kanchan Lata Singh. मैम आप स्क्रीन शेयर स्टॉप कर दीजिए. Dr. Kanchan Lata Singh.
Yes, ma'am.
Yes, ma'am. You can start.
My screen is visible.
Not yet. Present.
I'm visible.
मैम स्क्रीन विजिबल हो रही है बट आपको जहाँ पे जिस प्रेजेंटेशन को दिखाना है वहां पे जाइए, प्रेजेंटेशन पे जाइए जहाँ दिखाना है.
मैम visible. Morning, good afternoon, everyone. First of all, I would like to thank you for giving me this opportunity. I am Dr. Sanjay. Today I am going to present one of my research papers. Title of my paper is Physicochemical Characterization and Antimicrobial Activity of Mixed Ligand Complexes of Organic Third with 3 hydroxyphenyl, 5-order substituted phenyl pyrazolinates and 2N-salicylidyne 5-chlorobenzophenone. Now, what is the significance of organic metal? Organic substance that has been well known to both as a healer and a poisoner throughout history. It is potent neurological and liver toxic as well as lung, bladder and skin carcinogen. It has been a place in medical history, particularly in the treatment of 2 great sources of diseases in ancient time. First is sleeping sickness and the second one is great pox. A common concept in ancient Chinese medicine was to use a poison against a poison. Indian Ayurveda herbal medicine often contain arsenic, lead and mercury. Now come to the significance of the ligand. The ligand, we are chosen here 2 types of ligand because by aim is to synthesize the mixed ligand complexes. The first one is pyrazoline ligand, which is used in agriculture as catalyst, as a good agrochemical fungicides, as a potential antimicrobial agent. And the second one is 2N-salicylidyne 5-chlorobenzophenone, which is a Schiff base, which is very effective in constructing supramolecular architectures, strong antibacterial and antifungal agent, and as a potential anticancerous drug. This is the preparation method of the ligand. This is the dichloroarsenic third 3,2-hydroxyphenyl 5,4- substituted phenyl pyrazolinates, and this is mixed ligand complexes. All the mixed ligand complexes have been characterized by elemental analysis, molecular weight measurement, and spectral studies. All the synthesized compounds have also been screened for their antibacterial, antifungal, and antiviral activity. This is the fragmentation pattern of one of the compound. This is 1-hydrogen-13C and Fab mass spectral patterns of one of the compound. This is the antifungal and antibacterial activity. The slide 4 shows the mixed ligand complexes. Here, from the slide, it is clear that the synthesized compound have been found to be very effective antifungal agent. And here, in the antibacterial activity, this is
the mixed ligand complexes, disc number 5, which is also shows that it is also a good antibacterial agent. Now, this is antiviral activity. This data shows the inhibition zone of the compound. Now come to the conclusion. It can be concluded that the complexation of substituted arsenic third moiety with biologically active pyrazolone ligand result in increased activity of these complexes. From the antimicrobial data, it appears that when chlorine in chloroarsenic third dipyrazolinates is replaced by 2N-salicylidene 5-chlorobenzophenone moiety, there is no significant change, very slight significant change in antimicrobial activity of these complexes. This further confirms that the antimicrobial activity of these complexes arises from the pyrazoline moiety, but not from their CIPS-based moiety. But the 2N-salicylidene 5-chlorobenzophenone moiety does not affect the antimicrobial potential of the pyrazoline ligand. Here, the freshwater catfish, Heteronistis fossilis, are also exposed to arsenic third complexes. The concentration used are 2, 4, and 8 milligram per liter. After completion of the experiment, all the fishes subjected are active and live. This shows that the compounds of arsenic third has no morphological ill effect if used in aquatic environment. Thus, the synthesized compound may be used as a drug for fishes. However, the histological effect on liver, brain, and kidney tissues are under investigation. Correlation between geometry and around the central metal atom in the complex and antimicrobial activity of the same complex is a further area of the research. Thank you.
Thank you. Thank you.
Next, Upasana Yadav.
Alka Pandey, ma'am, can I present myself? Ma'am, Alka Pandey.
Okay, आप करिए तब. अलका पांडे, मैम अलका पांडे sharing.
Okay. Okay.
Kanchan ma'am, kindly stop your sharing. From the meeting tab, stop sharing. Yeah, click on stop share. Alka Pandey.
Number 6.
Yes, ma'am. After Alka Pandey, Upasana Yadav.
Ma'am, are you able to see my slides?
Yes.
Okay, thank you, ma'am. Good afternoon, ma'am, everyone. First of all, thank you for giving me this opportunity.
It's Ayushi Pandey's slide.
Sorry, ma'am. Sorry, ma'am. Stop sharing. ना शेयर करो बेटा. Ma'am, are you able to see my slides?
No. Please start immediately.
Sorry, ma'am. Share tasking. Share. Okay. Share. Sorry, ma'am. Good afternoon, everyone. I am Alka Pandey from Chemistry Department of Amity University. My topic is synthesis of novel pharmaceutical benzimidazole with green chemistry approach. The chemical synthesis in our environment is increasing. Advancement in chemistry have led to the production of medicines to reduce our problems. These same advancements have also generated various toxic chemicals, just like insecticide which dissolve in water. Not only these are harmful to our health, but also damaging to our environment, which result in destroying the food crops and pollute the ocean, due to which loss of biodiversity occur. Most of them are non-biodegradable. These non-degradable chemicals produces various types of pollutions which causes adverse effect on our health. To diminish the possibilities of these type of pollution and adverse effects, the green chemistry is an approach which aims to reduce the unused and generation of the hazardous substances by using the better chemical reactions. Specifically, the goal of the green chemistry is making the medicines or any other products less toxic. Heterocyclic compounds are present in a large amount in the biomolecules, and the heterocyclic compounds found a key structure in the medicinal chemistry with the extensive varieties of the pharmacological activities. Benzimidazole is considered as a potential bioactive heterocyclic compound exhibiting the various pharmacological activities. Benzimidazole is a bicyclic compound in which benzene ring is fused with the imidazole ring. It is a very important pharmacophore and the privileged structure in the medicinal chemistry. Benzimidazole is known for their several pharmacological applications, namely anti-cancer, anti-diabetic, anti-inflammatory, and anti-HIV and anti-fungal. Because of its valuable properties, the process of their synthesis have become the focus for the synthetic chemists. Nowadays, the most of the chemists adopt the greener way for the organic synthesis, which reduces the toxic substances in the environment, maximize the yield of the product. So during the preparation of benzimidazole and its derivative, we use the ionic liquid. Because of the ionic liquid is a green solvent, but the conventional method synthesize needed more heating time and apparatus setup. Green chemistry is a program suppose the invention of more environmental friendly chemical processes which reduce the eliminate the generation of the hazardous substances, minimize the hazardous products. Green chemistry is also the sustainable chemistry. Some of the principle of green chemistry is the prevention of waste, maximize the atom economy, less hazardous chemical synthesis, safer chemicals and the products used, increase the energy efficiency, use the catalyst, minimize the potential for renewable feedstock, real-time analysis, safer solvent, and hazardous accidents prevention. My main focus upon during my topic is the safer solvent to use. Prevention is a better. Green solvents are liquefiable gases, some of the bio-based solvent, water, ionic liquids. Green solvent are very important component of the any reaction, so we use the green solvent because the green solvent are the solvent which is environment friendly. Some of the green solvent are ionic supercritical liquid, CO2, water, bio-based solvent. Ionic liquid. Water is adaptable as a green solvent in many ways for carrying out the various organic reaction. Is a medium now of great interest because it is the most economical and bioacceptable. Supercritical CO2 have many of these properties: non-flammable, non-toxic, renewable, easy to remove from a product, do not contribute to smoke, low viscosity than water, inexpensive solvent, and fast drying time.
Please conclude your.
Okay, ma'am. Okay, ma'am. Ionic liquid are the type of the salts which consist of the organic cation and inorganic anion. The liquid room temperature, they have a cation, mainly alkylated imidazole, pyrrole, and the pyrimidine derivatives.
Kindly conclude, ma'am.
Okay, ma'am. Green chemistry is an approach by which the increases its yield, safe, and uses less harmful solvent for the environment have been reported in that paper, which may provide a useful route for the drug discovery.
Thank you.
Thank you, ma'am.
Next, Upasana Yadav.
Yes, ma'am, I'm sharing my slides.
Okay.
Can you see my slides?
Yes.
Yes.
Okay. So my topic was healthy habits, disease prevention and control of COVID-19. So it's easy to learn the bad habit, but it's very difficult to switch that back. The issue of the healthy lifestyle is very serious, but the people take it very lightly. Often it is seen that the people take steps to improve their lifestyle, but due to lack of determination, they quit in the midway.
Moreover, for a healthy lifestyle, it is important that you take a small step and one step at a time. And also do not go overboard with it. Besides this, healthy lifestyle will help you in life in a lot of ways. So for the healthy lifestyle, it is very important that you have a healthy eating habits. So base your meals on the starchy carbohydrates, eat a lot of fruits and vegetables, and eat more, for example, if you are eating the non-veg, that fish, including the portion of oily fish, then cut down on the saturated fat.
You can also see your slides. It is visible. Now it is visible.
Oh, sorry, ma'am. Again, I am sharing the screen. Wait a minute. Wait a minute, ma'am. I'm sharing the screen. Can you see my screen now?
No.
Now can you see?
Yes.
Okay. So do exercise regularly, avoid mobile. The biggest drawback in the generation is that they are obsessed with their mobile phones. That is causing the physical and mental problems. Connecting the positive mind, because more you indulge with these people, less you will go to the negative sides. Benefits of the healthy lifestyle. A healthy lifestyle has many benefits, not only for the body but for the mind too. To sum it up, we can say that there are various benefits of living healthy lifestyle. Besides, it improves the relationship in the family, but most importantly, a person who lives in a healthy lifestyle lives longer as compared to those who do not. Now, disease prevention. Disease prevention is a procedure that individuals adopt, particularly those with the risk factor of diseases, are treated in order to prevent the disease from occurring. Treatment normally begins before the signs and the symptoms of the disease occur or shortly thereafter. Treatment can include patient's education, lifestyle.
Again, your slides are not changing. Just conclude.
Okay, ma'am. So level of prevention are primary infection, primary prevention, then secondary prevention and tertiary prevention. Now, prevention of COVID-19. So standard methods of COVID-19: hand hygiene, personal protective equipment, respiratory hygiene and cough etiquette, cleaning and disinfection of devices and environmental surfaces, safe injection practices, medication storage and handling, waste disposal. So, ma'am, can you not see figure? I'm showing.
No, you are not changing slides. Just fifth number slide is there, but you should
wait a minute, ma'am. I'm stop. Can you see now the slides?
Yes, I can see.
Now, ma'am. Okay, ma'am. I am not slideshow mode पे मैं नहीं on करती हूँ फिर इसको. तो ऐसे ही दिखा. So, ma'am, this figure
जल्दी
conclude करिए.
Okay, ma'am. So there is a very important just now the concept of provision of safe water. The provision of safe water, sanitation and waste management and hygienic condition is essential for preventing the humans from the COVID-19 infection. There are evidences that show that the waste management practices from the homes, schools, marketplaces, healthcare facilities will help to prevent the human-to-human transmission and pathogenesis, including SARS-CoV-2 and the viruses that cause COVID-19. So, lastly, I would like to say that the protecting the sources of water and treating the water at the point of distribution and collection or consumption, and ensuring that the treated water is safely stored in the home, regularly clean and covered containers. Such measures can be effectively planned and implemented, monitored using the water safety plans. Conventional and centralized water treatment methods that utilize filtration and disinfection should be significantly used to reduce the concentration of SARS-CoV-2. Other human coronavirus have been shown to be sensitive to chlorination and disinfection with UV light. Thank you, ma'am.
Okay. Thank you.
Thank you, Pasna. Ma'am, Ayushi.
Okay.
Ayushi. I think she is not present. R.R. Avasthi?
Yes, ma'am.
Okay.
I'm present here.
Okay, sir. So kindly share your screen.
Yes, yes.
सर हो गया?
Okay. Yeah, my
वो
slide is
स्लाइड
visible.
शो पे ना करिएगा, स्लाइड
यस
शो पे
सर,
नहीं
उपासना
देखिएगा।
मैम म्यूट हो जाइए।
हाँ, वो वाला 1 2 3 वाला जो पीपीटी आता है नॉर्मल स्लाइड शो।
My slide, yeah, slide is
यस
visible.
सर, यस सर।
Okay. So good afternoon, every faculty members and present participants here. Myself, Dr. Avasti, and I am also faculty members of Navyuga PG College, Department of Physics. My topic is the structural topographical investigation of functional nanomaterials, and these are the outlines, which is the introduction effect of doping. That is, we are using the calcium ions and as well as inling temperatures. And the material problem exists in literature. There are various problems when we are synthesize the nanomaterials. The main objective of the present investigation and VR methodology use and application, and what is the characterization we have done, and then the outcome of this. So we are working on the multiferroic nanomaterials, generally basically bismuth ferrite, and is the crystal structure of bismuth ferrites. You are clearly showing that the diagonal elements is iron, and bismuth is come to the corner and face centered at oxygen ions. So the formation of this bismuth ferrite crystal structure is that, and due to the distortion of diagonal elements, this the polarization we have obtained at according to the diagonals 110 planes. And so the multiferroic, generally we are working on multiferroic. So basically the multi means two or more than two properties of materials are called multiferroics. So we are know all about the ferroelectric and ferromagnetic. These two properties are exist in the similar materials. So not only exist, but it is coupled, and it is very important because the ferroelectricity we are know about the ferroelectricity can be use the electric polarization are obtained when use the electric field, and the magnetization can be obtained when use the magnetic field. But in magnetoelectric materials we are use the electric polarization can be controlled by the magnetic field, and magnetization can be controlled by the electric field. That is why the cross coupling of electric and magnetic field is exist in that materials. That is why it is a very important for electric memory devices. We are using the calcium doping concentration, and we are found that the surface properties as well as electrical, magnetic and dielectric properties of the materials is visually improved, and the antiferromagnetic to ferromagnetic transition we are also found in that materials. So problem existence basically the high leakage current density at a room temperature. So we are want to improve that high leakage current density, and the crystal symmetry is rhombohedral distorted, and the incommensurate long wavelength cycloid spin structure. Due to that, microscopic magnetizations is poor. So it is shows the
Slides are
antiferromagnetic
not
character. So we are
properly.
want to improve these two
Yes, sir.
properties, which is hinders
Slides are not properly
potential
displayed.
application of that materials. So we are clearly shows that the bismuth ferrite crystal structure of polycrystalline materials from XRD diffraction pattern. There are some impurity phases Bi2 Fe409 as clearly shows by sticks, and when we increase the calcium ion dopant concentration, then the stick that is the impurity phases is diminished, and as well as 104 and 110 peak is merged together.
Please
and
conclude your
the one
findings.
single phases. So that is the distorted
Please conclude your findings.
structure is converted into ortho.
Sir, kindly conclude your findings.
And here the polarization is clearly shows that the electric field is applied, and we have found that when increase the doping concentration, then the ferrohysteresis loop is also increases, and it is clearly shows. So we can use this material as a ferroelectric memory devices. When increase the calcium doping concentration, then there is a WS plot is clearly shows the strain of that material is increases when increase the doping concentration, because the crystallized size of that material is decreases. Here the AFM
Please
picture
conclude.
clearly shows that cubic crystal structure when we pure form of BFO, when
Dr.
increase
Awasthi,
the higher
kindly
doping
conclude it.
concentration, that is 15% of calcium, then the cubic structure is converted into a spherical like structure and very dense, because the particle size is also decreases with increase the doping concentration. Here same picture is also
Please
reveals
conclude.
the same mechanism.
Please
where
conclude
the
your
cubical
lecture.
structure is converted into a spherical like structure. There is the ring like structure is shows that the clear formation of nanoparticles of BFO nanomaterials is the time pictures of
Sir,
it is a
kindly
time
conclude
picture.
it.
There is also
Thank you.
clearly
Please
shows that
conclude.
homogeneous distribution of the
Please
particles
conclude your
in throughout.
findings.
Next, Sukriti Singh. Sukriti Singh? Okay. Aparna Mishra?
Yes, ma'am.
Ma'am, Sukriti.
Okay, Sukriti. Aparna, wait. Sukriti, you can present now.
Yes, ma'am.
Yes. Okay, but I want to conclude that. I want to conclude that. So the past. Okay.
The time is over, sir. So kindly.
Was 1 minute, ma'am. 1 minute. I want to conclude that. So the possible application of that materials is for magneto-electric memory devices. Here it is you are very well known, that is the integrated circuit, and here magnetic chip, and here it is when this material can be used for terabyte magnetic memory devices. Here we are using GB, gigabyte like, but that materials can be applied for terabyte magnetic memory devices. That is why it is a very important materials, because there's some hindrance properties. So we want to improve that properties, and we want to for application of terabyte memory devices. There are characterizations
Thank you, sir.
UGC. Okay, thank you.
Thank you, sir. Thank you, sir.
Sunita ma'am.
Yes, ma'am.
Here, Dr. Rekha Rani from our contact. And with due
Yes, ma'am.
permission of the chairperson, I am suggest to Dr. Awasthi, if somebody is saying to you again and again, please stop. Why you are not listening? You are the senior faculty in the Navyuga. Please listen to this. It is not giving a good impression to all of us. Thank you very much. Thank you, madam.
Thank you, ma'am. Next, Sukriti Singh. Please screen share kariye.
Ma'am, my slides are visible?
Not yet.
Okay, just a second, ma'am.
And it's a request. Please आप लोग टाइम का ध्यान रखिए. बार-बार इंटरप्शन अच्छा नहीं लग रहा है.
Ma'am, is my slide visible now?
Yes.
Okay. Good afternoon to one and all present here. Myself, Sukriti Singh from Amity University. And the topic for my presentation is green synthesis of copper nanoparticles using top to bottom method through enzymatic activity in mulberry leaves. I'll come to the introduction, where nanoparticles are very small particles that range between 1 to 100 nanometers in size, and same goes for the copper nanoparticles that are copper-based particles that also range from 1 to 100 nanometer in size. The copper nanoparticles can be formed by the natural processes or through the chemical synthesis. The copper oxide nanoparticles have black brownish powder. They are graded harmful to humans and as dangerous for the environment, with the adverse effect on aquatic life also. Next is the review of literature, where nanotechnology is the scientific revolution of the 21st century.
Just you go through this. Next.
Okay, ma'am. Next is the work which I have done with the Amity University, which is the materials and methods. The materials required are the mulberry leaves, the media, the chemicals that are yeast extract, peptone, glucose, malt extract, etc. The methods for the top to bottom method and the enzymatic activity, which first includes the preparation of the culture media and the yeast inoculation, which is done under 30 degrees Celsius in a shaker incubator for over 24 hours. Next is the collection of leaf sample and the leaf extract preparation. The leaf sample was collected from the university itself, and the extract preparation was done using the tris HCl buffer and copper sulfate solution. And these were kept for boiling and then after centrifuge. Then for the procedure was done for continuous 6 days for identifying the and matching the graph peaks. The scanning was done at 200 to 800 nanometer on the spectrophotometer. Now to perform the MTT assay test for the leaf sample and the leaf extracts, the media was prepared, and the MTT, which is I think it is it is the artificial MTT, and it is the MTT is read at 570 nanometer, and the shaker incubator is also used for performing the MTT assay test. Then for identifying the cell growth with the copper nanoparticle interaction, the conical flasks were used, the YPD media and the flask were used. The copper nanoparticles were taken and they were incubated respectively for 1 day at 30 degrees Celsius, and the OD was read at 600 nanometer after 1 day. Then the standard identification of ferricyanide using the buffer and ferricyanide at different concentrations. The standard identification was got at 200 microliter ferricyanide.
Now you come to your conclusion, please.
Okay, ma'am. Okay, ma'am. Hereby, I conclude that the spectrophotometric and color change method showed that the copper nanoparticles were synthesized after 1 week. These are the results, what the study we have done. The conclusion says that the spectrophotometer changing method was showed the copper nanoparticles were synthesized after 1 week. The effect of green synthesized copper nanoparticle was tested on the cell viability by the MTT assay in Saccharomyces cerevisiae, and as the results showed the 77.42% decrease in the mitochondrial dehydrogenase activity, and on the contrary, the commercially available copper oxide nanoparticle, which is from Sigma Aldrich nanoparticle, showed the 2.5% increase in the cell viability. Although we have observed no significant effect in the transplasma membrane electron transport, which is TPMET, with both the commercially available and green synthesized copper nanoparticles. Thank you, ma'am.
Okay. Thank you. Sukriti, have you conducted all these experiments?
Yes, ma'am. These all experiments were conducted, and within 2 months, we have got some results.
Okay. Okay. Thank you. Nice work.
Thank you, ma'am.
Next, Aparna Mishra.
Yes, ma'am.
Yes, you can share.
Am I audible?
Yes, you are.
A computer would deserve to be called intelligent if it could deceive a human into believing that it was a human. With this quote of Alan Turing, a very good afternoon to all the respected members and my colleagues. In this world of crypto metaverse, I, Aparna Mishra, BSc third year student from Navyuga Mahavidyalaya, Lucknow, is going to explain what is happening in the biomimetic world. The fundamental principle of neuroscience is the integration of anatomy, molecular biology, psychology to explain the action and the working of neurons and neural circuits. Neuroscience has now reached a stage where it can document the communication among neurons. It's now possible for people suffering from any physical impairment during any neurological disorders. Our thoughts and actions are nothing less than electrochemical impulses. According to a well-known entrepreneur, Elon Musk, we can intercept brain signals, record, and alter them to enhance the brain activity. In the ocean of biomimetic, my topic is Neuralink. Neuralink is a computer body interface, which means that it links your actual body
It simply means that it links your actual biology of your body to computers, and using those computers you can actually send electric signals within our brain, because whenever we do movements is basically some sort of electricity that is going on in our body. Neuralink is supposed to fix lot of problems, but the concern that some coder go crazy and just put that in your brain is good for humankind. Well, when cars were invented, there was a possibility of accidents. When social media was invented, there was a question of privacy. But now we all voluntarily enjoying the both. Well, it is not practically possible for human to install Neuralinks because this surgery is complex in many ways. Therefore,
slides are not changing. Your slides are not changing.
Ma'am, actually, it's a poster related to Neuralink.
Okay.
Well, it is not practically possible for a human to install Neuralink because this surgery is complex in many ways. Therefore, specially designed robots are ensured that the electrodes implanted correctly without damaging the blood vessels or any other arteries. In fact, there is a company named Synchron implanted a brain chip into the jugular vein of someone called Mr. Philip, who was paralyzed from many years. And after that, he took only 4 hours to restart his work and take control over the device. Even after inserting the Neuralink, he tweeted.
Aparna, kindly conclude.
Yes, ma'am. I would like to conclude with Neuralink is the future technology that can heal multiple disorders, and at some point, second or third version of Neuralink can help to enhance the brain with super memory. But biomagnetic world has more ambitions from this innovation, such as reading and writing memories and emotions, eventually combining human intelligence and the artificial intelligence for better brain performances. But it only replicates the basic movements. Can a Neuralink perform complex processing of brain activity? For now, it is impossible to say more about it. Thank you, everyone.
Okay, thank you.
Ma'am, if there is any question, I am open.
Okay. Thank you, Aparna. Next, Dr. Dhondi Ram Tukaram Sakhare. Sir, are you there? I think not.
Excuse me, my Dr. Rajan Kumar. May I present?
One second, sir. राजेंद्र कुमार जी, आप थोड़ा वेट कर लीजिए अभी.
ओके मैम.
ठीक है.
Please start your lecture.
Hare sir, are you there? I think कुछ दिक्कत है उनके साइड से. सर आप एक बार फिर से चेक कर लीजिए, तब तक मैं डॉक्टर रश्मि गुप्ता. नहीं. डॉक्टर अलका गुप्ता.
I am very much
yes,
here. I am here. Please.
Okay, मैम आपने कुछ बोला नहीं. डॉक्टर रश्मि.
Yes. I am stuck somewhere. I am without present paper with my phone only. Okay, I'm sharing my screen. Yes. Is my screen visible?
Yes.
Yes.
Okay. Yes. My paper focuses on wastewater treatment, management, and possible use of treated water for various applications. Water quantity and quality is of vital importance for the survival, sustainability of economic, society, and environment. Maintaining adequate supply of clean water is one of the most significant challenges of the 21st century. The shortage of water is further aggravated by insufficient treatment of water and wastewater, particularly with rapid population growth and urbanization. As we can see in the second slide, that different sectors use different quantity of water. In India, as we can see, in the year 2000, irrigation sector used 5 billion cubic water.
Slides नहीं दिख रही हैं.
Slides are not. Okay. Something, ma'am. I have no up here. How now? Nothing is visible.
No.
How it happened?
एक बार reshare करिए.
Okay. I am trying.
Or maybe she can oral presentation with due of the chair.
Okay. Someone else is. Okay, there is.
Yeah, someone else has started sharing. That's why your slides are not seen.
Okay.
Okay.
I don't know what happened. Sorry, I'm not used to my
laptop.
Actually, I'm somewhere in the hospital. Okay, thank you. As we know, that shortage of quality water is positive to human health and well-being and most of our productive ecosystems. Water resources are, but the amount of water produced is increasing. Wastewater produced and the infrastructure and systems are not adequate for this increased volume. Industrial agricultural waste is discharged into water bodies, which contaminates fresh water and coastal, threatening food security and bathing water, and a major environmental management. Domestic water is a major source of water pollution. Poor rising population have resulted in illegal settlements and slums where sanitization services are poor, inadequate, and unreliable. Treatment. The wastewater can by primary treatment, secondary treatment, primary treatment, remove the flow solids the wastewater. This physical liquid separation is a mechanical process, although can be used sometimes to accelerate the sedimentation process. Price of grit removal and screening, sedimentation and flotation followed by filtration. The secondary treatment helps eliminate the dissolved organic matter that escapes primary treatment. Tertiary treatment process ensures nearly 99% of all impurities and are removed from water. To make the treated water safe for drinking purposes, water is treated individually or in combination with.
We just conclude within 1 minute.
Okay. Different microbes like Pseudomonas, Bacillus, Azotobacter, Pastorella, Xanthomonas are used to treat the wastewater. Water reuse often offers promising opportunities to reduce water stress and to increase available water resources for different purposes ranging from agricultural use, industrial process to direct potable use. And to conclude, Due to high consumption of fresh water along with its shortage afflicting many parts of the country, the pressure to recycle and reuse wastewater is constantly increasing. As such, the planner should evaluate the potential use of wastewater for sustainable development. Advanced technologies should be used in place of conventional technologies to minimize water loss and provide high quality water that can be recycled and reused. Thank you.
Okay, thank you. Rashmi ji.
Okay, thank you.
Next.
Dr. Sakhare.
Yes, Dr. Sakhare. डॉक्टर सखारे आप हैं? ओके मैम, नेक्स्ट पार्टिसिपेंट को बुलाएं?
हाँ बुला लीजिए।
डॉक्टर अलका गुप्ता।
Yes, ma'am. I am presenting my screen.
Okay.
Is my screen is visible, ma'am?
Yes.
Good afternoon, everyone. Myself, Dr. Alka Gupta, Department of Chemistry, Brahmanand College, Kanpur. My topic is conserve water, food and energy for future. The water should be of sufficient quantity to enable sustainably livelihood and socio-economic development and be managed to avoid waterborne pollution and disasters as well as preserve water-related ecosystems. It's the reason states and countries with the rapid urbanization, population growth, society has with its waste has become necessary to adopt some new technology to make the best use of waste. Fiber and food on lands, water, energy and other resources. Increased food production is impossible without extra energy inputs. Energy costs are also an important cost factor in agriculture. There are so many factors leading insecurity in future, such as there can be several causes of water scarcity, including low rainfall, climate change, high population density, and over-allocation of a water source. There are many overlapping terms used in discussion of water insecurity, such as water contamination, water stress, water scarcity, and so many. Land grabbing is another factor when land traditionally owned or farmed by families or groups or communities is taken away from them by large investors or influencing government officials. They acquire the land to expand their empires, extract natural resources, or even grow food for export. Some of the resources that people use to generate electricity are finite. The planet is not capable of creating additional coal or uranium supplies. So there are various approaches for conservation of water, food and energy, such as global investment in water infrastructure, management of water resource diversity protection, and human. The structural diagram of non-waste technology required to be improved and should consist of a linking up of several different industrial, agricultural activities in such a manner that the waste of one industry become the raw material of another. Such technology will depend on multiple uses of resources and internal transfer of waste from one sector to become the raw material for another. Being a global challenge, governments and international institutions have employed all the possible means to defeat food insecurity. Governments at the national, regional, and local level may implement policies to promote energy efficiency, So overall, good water quality is essential for the sustainable development of society to support human health, ecosystem services, and agriculture and industrial production. Good management of water resources can jointly manage biodiversity, protection, and human waste water security. It is achieved when there is enough water for everyone in a region and the water supply is not at risk of disappearing. A community is composed of academics, practitioners, and policymakers from both the water and nutrition communities. This group exchanges ideas on cross-sectoral linkages, develop research and insights, and aim to advance policy action. So overall, on a global level, energy use can also be reduced by stabilization of population growth. Basically, energy conservation and can make this a better world both today and in the future. Thank you, ma'am.
Thank you. Thank you, Dr. Alka Gupta.
Okay, thank you, ma'am.
थैंक यू मैम. नेक्स्ट सखारे सर, आपको प्रेजेंट करना है. मैम वो बाबा स्क्रीन शेयर कर रहे हैं बट आवाज नहीं आ रही.
स्क्रीन तो शेयर हो गया.
यस मैम.
डॉक्टर सखारे.
Dr. Sakhare, you are not audible.
Dr. Sakhare? Just
Dr. Sakhare, your mic is muted.
Even slides are also not properly.
Yeah, they are not moving.
Yeah.
Dr. Sakhare, stop your presentation, then I will call next. Atul Pandey.
My screen is visible?
Yes.
Not yet.
Not visible.
Visible?
No.
Good afternoon to all of viewers. My research topic, effect of pollution on river Narmada. Myself Atul Pandey from Indira Gandhi National Tribal University, Amarkantak, Madhya Pradesh. Water is a precious liquid for any living beings which lies in earth. India is the country of river, and my specific topic in Narmada. So Narmada is also known as a lifeline of Madhya Pradesh, Central India, and flow through state of Madhya Pradesh, Maharashtra, and
Those
Gujarat.
slides are not visible. You are visible, but not your slides.
Sakhare sir, stop presenting.
Huh?
नेहा मैम.
आप बोलिए. यस मैम.
मेरे एब्स्ट्रैक्ट ना, जो आपकी बुक है एब्स्ट्रैक्ट वाली, उसमें है नहीं. मैंने
मैम हम वो चेक कर लेंगे, अपने एंड पे ठीक करके आपको भेज देंगे. अगर नहीं होगा तो आप परेशान मत होइए.
That we will discuss later on. If it is not there, that will be rectified.
अतुल सर, आपकी स्क्रीन शेयर नहीं हो रही अभी.
I think first we'll need to सखारे सर को जब हम रिमूव करेंगे तभी दूसरों की स्क्रीन शेयर हो पाएगी. अदरवाइज वो खुद प्रेजेंट करें.
हाँ मैम, वो बोल भी नहीं रहे कुछ, बस स्क्रीन शेयर.
तो अभी फॉर द टाइम बिंग उनको रिमूव कर दीजिए मीटिंग से, ताकि अगले लोग प्रेजेंटेशन कर सके.
नेहा मैम, सुनील जी को बोलिए उनको रिमूव करें. ओके. हमारे एंड से नहीं होंगे.
I'll just do. Yeah.
My slide is visible?
Yes. It's not slides. You are visible. Share your screen.
Yes, madam. Visual, madam?
Yeah,
Yes.
now it's
Now visible.
Water is precious liquid for any living beings which lies in earth. India is the country of river, in which my specific topic in Narmada River. Narmada River is also known as lifeline of Madhya Pradesh in central India and flow through the state of Madhya Pradesh, Maharashtra and Gujarat. In river, fishes have been considered as a bioindicator, mainly hilsa, which is found in a hill area, to observe and understand in various freshwater ecosystem. Fishes are vital for biodiversity perspective since they live in various ecosystems. Narmada River also a very big and rich biodiversity of fishes. Or in sometimes Narmada River get more polluted and various pressure on the riverian ecosystem is increasing in the specific in Amarkantak region due to high anthropogenic activity. The growth of diversity of aquatic fauna, especially the ichthyofauna of the riverian ecosystem, is highly influenced by changing water quality like COD, BOD, oxygen, and limnological parameter. I found in my current research heavy metal like arsenic and aluminum, which is very harmful for human beings and also for fishes. When fishes eaten by tribal people and other human beings, they are getting cardiac arrest and neurological diseases. So I found 20 mg per liter 20 mg per liter aluminum, which is found in Amarkantak region. So this is very harmful for our human being. And also 3 mg per liter. So this is very hazard for human being. Thank you.
Okay,
thank you, Dr. Atul. Before calling Dr. Harendra Pal, Dr. Rajendra Kumar sir, आपका है तैयार?
Yes, ma'am. I am sharing just now.
Yes, ma'am. Dr. Rajendra Kumar room में
Third presentation.
Yes, ma'am.
Third number. Okay.
मैम स्क्रीन शेयर नहीं हो रहा है इसमें से ये एक टैब बेस्ट फॉर वीडियो एंड प्रेजेंट स्क्रीन
शेयर करिए।
मैं यहाँ से कर रहा हूँ, इसमें 3 ऑप्शन दे रहे हैं, योर इंटायर स्क्रीन, योर विंडो, ये टैब, ये किस में से जाएगा मैम? बेस्ट फॉर वीडियो एंड
आप अपनी प्रेजेंटेशन है तो मेल से
ये एक्चुअली ये एंटायर स्क्रीन से कर दीजिए और अपनी गूगल मीट को मिनिमाइज करके अपनी प्रेजेंटेशन पर चले जाइए, हो जाएगा।
पहले से प्रेजेंटेशन रखिए।
ये एंटायर पेज प्रॉब्लम आ रही है, शेयर शो नहीं कर रहा।
सर आप एक बार और रिजॉइन करें, तब तक मैं नेक्स्ट पार्टिसिपेंट लाते हैं।
ओके ओके मैम,
ठीक है। डॉक्टर हरेंद्र पाल।
Yes, good afternoon all of you.
Yes.
I'm sharing my screen.
Okay.
Visible? Is someone comfortable?
No. Okay, yes.
Okay. So mechanics of in our daily life. Department of Mathematics from College Bahedi. But is the main name of rotating. In this paper, we have to also prove all the results that are valid even in case of fluids and solutions have been optics and radial flows. These are some daily life use. Except also things like this is flags, making coffee, showering. This concept is in all the formulas which is the equation of continuity. Second one is the equation of
it's not visible.
Is it visible?
No.
Wait. Visible?
Yes.
So I was telling about these making coffee. These concepts are used in daily basis in these things. So these are the formulas which I have derived. This equation of continuity. Second one is equation of solenoidal. And these are the other results which I have come across. I have mentioned all of these in my paper. In last, these are the radical flows equations. And at last, it is vortex flow. So, and at last, these are the references. Thank you, that's all.
Okay, thank you.
Thank you, Dr. Haren. Next, Dr. Savita Agrawal.
Yes, ma'am.
Yes, you can share your link.
Ma'am, I have oral presentation.
Okay, ठीक.
Good afternoon, everyone. Today, I, Dr. Savita Agrawal, Assistant Professor, Gokuldas Hindu Girls College, is here. My topic is a study on potential effect of chemicals on human health. As in the modern. Yes, ma'am.
Share your screen.
Ma'am, I have a oral presentation. As in the modern era, everything is made up of chemicals, and also chemicals has made our life easier. With the development of new technique and use of chemicals, we have manufactured different polymers, dyes, paints, drugs, and so on. But mostly all the chemicals are toxic, and toxicity depends on the dose of chemicals and also the age of exposure. This chemical exposure is high to the workers who works in the chemical industries, coming down with certain diseases. Approximately 100,000 synthetic organic chemicals are present in the global market. There is very high risk in distribution, handling, carrying chemicals. It can be understood from the fact, some fact I have taken. On 7th May 2020, 11 people died and more than 800 people fall severely ill due to the exposure of poisonous gas, styrene, from LG Polymer Industry, Visakhapatnam, Andhra Pradesh. And 9th October 2018, 9 people died and 14 people got severely injured due to the explosion of gas pipeline in Bhilai steel plant, Chhattisgarh. And many more are also there. So globally, it is essential to protect the workers who are involved directly in these activities. Toxicants can also be entered into our homes, workplace, and store places from everyday products we use, in the form of phthalate, bisphenol, xylene, methylene chloride, dioxins, vinyl chloride, etc. We are exposed directly or indirectly to these chemicals by the food we eat, water we drink, and air we breathe. Some chemicals originated from occupational exposure. Us are exposed to chemicals that are present in hair dye, such as paraphenylenediamine, resorcinol, lead acetate, and toluene, etc. All these chemicals are associated with a number of health issues like multiple forms of cancer, eye injury, allergic reactions, hormonal endocrine disorders, birth defects, mental disorders. Exposure of chemicals is area of concern. We need to be aware of the consequences and remedies. Therefore, the study aims to examine the potential effects and consequences of chemicals on human health. Thank you, ma'am.
Thank you, Dr. Savita. Next, Bushra Yasin. Savita ma'am, mute हो जाइए. Bushra Yasin. Next, Shalini Agrawal.
She's absent.
Okay. Next, okay. Renuka Devi.
हेलो मैम, एक्चुअली मैं बुशरा यासीन ट्राई कर रही है पर उनका अनम्यूट नहीं हो रहा है.
ये तो उनके एंड से उनको देखना होगा. बुशरा यासीन को फिर से बोलिए रिजॉइन करने को.
Yes, ma'am. Yes, ma'am. Am I audible?
Yes.
Okay.
Kindly share your screen. Bushra Yasin.
यार इसमें Yes, now.
Yes.
Now Bushra. Yes, ma'am. Okay. A very good afternoon to all the teachers present over here. I am a research scholar, Bushra Yasin, from the Department of Chemistry, University of Lucknow. The title in my presentation topic is Synthesis of Silver Nanoparticle by Chemical Reduction Method in Presence of a Neuroleptic Agent and its Characterization. So in this, I have synthesized silver nanoparticle by using a gabapentin, which is a medicine for the reduction of silver nitrate into silver nanoparticle. GBP, why I have chosen? Because it is environment friendly, it's a medicine and having a chemical formula C9H17NO2, and it is also known as cyclohexylic acid. This drug is an anticonvulsant and use various psychiatric as well as migraine disorder, and is also used in the treatment of bipolar disorder, mood stabilizer, and neuropathic pain. The important key of using this medicine because it is soluble in at all pH, whether it is acidic, basic, or alkaline, and it has found that at physiological pH it acts as both reducing as well as stabilizing agent. So by using this medicine, there is no need of stabilizer for the preparation of nanoparticle. So this is the flowchart representation of my work plan, how I have prepared and characterized. So initially all the solution, this reaction was performed at 40 degrees Celsius. So all the chemicals which is used for this preparation has been equilibrated at 40 degrees centigrade, and then the gabapentin and NaOH was mixed together, and then this solution was added to AgNO3, and after the addition the stock solution was prepared, which was stirred for an hour. After stirring, the solution was centrifuged at the same temperature for an hour, and after centrifugation at the bottom we got a powdered sample which was washed several times to remove the other materials present in this mixture. And after this the sample was dried in oven for an overnight, and that dried sample was subjected to characterization. These characterization techniques were UV, FTIR, PX, EDX, SEMTEM, which is used to confirm the presence of silver nanoparticles. So this is the graphical representation of the formation of used for the formation of nanoparticle. So initially gabapentin and NaOH was used, and to this
AgNO3. This AgNO3 was colorless in nature, and when silver nanoparticle was formed, this was the color of the solution was changed from colorless to pale yellow, and after 100 minutes it was found that a bright yellow color was appeared, and after a month when the solution was kept, the brown color solution was obtained. So the intensity of the solution was changes with time. The first one, if you see, there are 3 graphs. At 200 nanometer, it is the pure gabapentin peak. This is the UV analysis. And then at 415 nanometer, silver nanoparticle formation. This is the SPR range of silver nanoparticle. The second graph is the EDX analysis, which confirms that in our sample, silver was present in elemental state. And then in the last graph shows the XRD analysis, which is used for the X-ray diffractometer, and this is used to confirm the crystalline nature of our sample. And from this, the graph which is formed intensity and 2 theta, this was the theta, the values related to theta was matched with the JCPDS file, and it confirms the formation of silver nanoparticle. And it also confirms that it is in HK, that it is crystalline and monodisperse. These are some other techniques which is used for the confirmation of nanoparticle. The first image A shows SEM and the B show TEM, which clearly shows that the formed nanoparticles are in spherical in shape and they are lightly agglomerated. So from SEM and TEM, we can confirm the morphology. The C diagram shows what is the average particle size of our nanoparticles. This shows that within the range of 5 to 25 nanometer, our particle size distribution was there, and the major particle size was from 20 to 30 nanometer. Now this is the summary of my work. Why I have chosen gabapentin? Because it acts as both reductant as well as a stabilizer, and this was prepared by chemical reduction method, and this method can be adopted because it is simple, economical, rapid, and this can be the application of my study can be used as useful in large scale synthesis of silver nanoparticles at industrial scale. And there is an interaction of the pharmaceutical drug gabapentin with silver nanoparticles. So in application, it can be used in drug loading capacity and drug delivery and diagnostic. Thank you, ma'am.
Okay, thank you.
The next participant is Renuka Devi. Renuka ji, are you here? The next participant is Zoya Amreen. Zoya Amreen. Next, Dr. Bhartendu Pati Tripathi. Dr. Bhartendu Pati Tripathi, are you here? Anurag Prasad.
Good afternoon, everyone. I'm sharing my presentation. Is it visible now?
No, you are visible.
Not yet, sir.
Is it visible now, ma'am?
No.
Yeah, now it's visible, Anurag ji.
Good afternoon, everyone. The topic of my presentation is use of vibrational microscopy as a valuable tool in analysis of pharmaceutical drugs. I am Anurag Prasad from MIT University, Lucknow. Aligning myself with the theme of this conference.
No, but the presentation is of somebody else. Just confirm.
Ma'am, is it visible?
No, no.
Ma'am, there's some unmute problem. I will be giving after 5 minutes. This is Zoya Amreen.
Can I present now?
But your slides are not visible. You are visible.
I hope it is visible now, ma'am.
Yes, now it is visible.
The topic of my presentation is use of vibrational microscopy as a valuable tool in analysis of pharmaceutical drugs. Now, I was saying, aligning myself with the theme of this conference, I would like to put the relevance of my topic. The rapidly changing requirements in pharmaceutical world need powerful tools and techniques which can enhance the process of drug discovery and development. And it was very evident in last few years also. Vibrational spectroscopy is a very powerful and valuable tool in the study of pharmaceutical compounds. It has shown great potential in this field in last few decades, leading to many technological advancements in pharmaceutical analysis. It is a fast and non-destructive technique. It is used in different ways as infrared spectroscopy, near-infrared spectroscopy, Raman spectroscopy. They can be used alone and also in combination with some other analytical techniques. Using vibrational spectroscopy, real-time in situ monitoring of phase transformations of samples can be achieved. It can probe intermolecular interactions between active pharmaceutical ingredients, and it can help in understanding the properties and mainly stability of these ingredients in the process of development, and later help in process formulation and formulation development. Specific chemical signatures of sample can be achieved with Raman spectroscopy, which can be utilized in the research of pharmaceutical crystals. With Raman spectroscopy, phase transitions, solid and liquid formulations can be studied non-destructively, which benefits further in development of drug and large-scale production. These techniques help in providing accurate analysis of essential information about the pharmaceutical drug under investigation. Development of simple-to-use and cost-effective spectroscopy instruments have led to an exponential growth of research and literature in this field. It is a very established and effective experimental technique for study and analysis of molecular chemical composition. It can help in determining precisely important parameters like bond angle, bond length, and bond distortion relationships, which can be useful in predicting the activity of pharmaceutically active compounds in drug development process, and hence it becomes a very useful technique in area of pharmaceutical science. It includes many techniques such as infrared and Raman spectroscopy. These methods are based on the study of changes in molecular vibrational state caused by the energy transferred by incident photon after the interaction of molecule with electromagnetic radiation. It is mainly applied to infrared identification of active pharmaceutical ingredients by comparing spectrally a sample against a reference standard data. So it is basically a study of vibration of chemical bonds. It can be used as absorption as well as emission spectroscopy. This is the setup of an infrared spectrometer. Raman spectra arises from change in polarizability of the molecule in off-resonance condition, and it is the inelastic scattering of photons of light after interaction with matter. It has a much lower profile with regularity authorities as compared to infrared methods. It is studied in form of Stokes Raman scattering, anti-Stokes Raman scattering, and relay scattering.
Please include your findings.
Sure.
Have you conducted these experiments?
I'm currently doing work in this field. So my idea was to put...
you have conducted these, or it is just a theoretical paper?
No, I'm currently working in this field, and this paper is about a review of these things so that the aspiring researchers can also get some help. Okay. So medical advances have helped millions of people live long, healthier lives. We hope these improvements to
Just to conclude,
for organizing such a wonderful conference. Thank you.
Okay, thank you so much.
The next participant is Pradeep Kumar Pandey.
Yes, ma'am.
Pradeep ji, are you there?
Screen share.
Kindly share your screen, Pradeep ji.
Yes, ma'am. ये पता नहीं क्या है।
Someone else is also presenting, I think.
मैम एक मिनट मैम स्क्रीन शेयर हो
Yes, yes.
A very good afternoon to all of you present here. I am Pradeep Kumar Pandey, PhD research scholar, Chemistry Department, University of Lucknow, under the supervision of Dr. Jas. Today my presentation topic is the albumin-mediated synthesis and characterization of zinc oxide nanoparticles for in vitro evaluation. Since there are so many methods for the synthesis of nanoparticle as a chemical method, physical method, and biological method, but I am adopted the biological method because it is a cost-effective and eco-friendly. The characterization of nanoparticles can be done by the using UV visible spectroscopy, FTIR, FSM, and TEM analysis. This is the result of my work by using the UV visible spectroscopy. The SPR range, the peak of this zinc oxide nanoparticles obtained at 374 nanometers. And this is the FTIR analysis. There is so many stretching frequencies, such as 3411 is the OH stretching frequency, and 2922 is the CH, and 606 is the zinc oxide stretching frequency, which confirms the formation of the zinc oxide nanoparticles. This is the powder X-ray diffraction studies. The high degree of crystallinity of the synthesized zinc oxide nanoparticle samples zinc oxide nanoparticles determined through the XRD analysis given, which shows powder XRD pattern of zinc oxide nanoparticle having the number of characteristic peaks 2 theta at the diffraction angles. This is the field emission scanning electron microscopy. The surface micrographs of the synthesized zinc oxide nanoparticle at different magnifications, such as the 500, 5000, and 1000 is shown. These efficiency images of zinc oxide nanoparticles show.
Hello, you are not audible.
Pradeep ji, you are not audible now. Pradeep ji, are you there? Kindly stop sharing if you're not there. What happened, Pradeep ji? The next participant is Nishat Afza.
Yes, I am here, ma'am.
Yeah.
audible?
Yes.
Kindly share your screen.
Imidazole derivative. In this poster, I have shown theoretical and experimental correlation of the entitled compound using DFT method B3LYP6310 basis set. Imidazole, a well-known heterocyclic moiety, exhibiting a wide variety of pharmaceutical application. Imidazole nucleus, another theoretically important moiety, find its broad application as anti-cancer. This is the optimized molecular structure of the entitled compound. This table one shows a good correlation between experimental and calculated and proton and C13 NMR data. This is the pictorial diagram of homo-LUMO energy. Homo-LUMO transition having energy 5.1997 electron volt. This is this diagram. This is the MESP plot of the title compound, which gives information about the electrophilic and nucleophilic site. This is the blue region shows. This blue region shows the more nucleophilic region, and blue region shows the more electrophilic region. The maximum negative electrostatic potential is situated around nitrogen 6 with MEP value around -7.352, and most positive region is found at C10 and C15, that is 7.352 AU. The present work comprises of synthesis and respective characterization of novel compound by spectral and computational method. Thank you, ma'am.
Ma'am, can I represent it? This is Zoya Amri.
Zoya, yeah, you can start now.
Okay, thank you, ma'am.
Zoya, please start.
Yeah, ma'am.
Share your screen.
Ma'am, I'm sharing, it's not coming. I'm trying. Ma'am, I think there's a technical problem. You can give to someone else. Thank you.
Anam Zadi. You please stop sharing, Zoya. Anand Zadi, are you? Dr. Casey Dubey.
Yes, ma'am, I'm here.
Anand Zadi, you are here. Yeah, please start now.
A very good afternoon to one and all. Myself, Anam Zedi. I'm here to present my paper. Is my screen visible now?
No, your slides are not visible. You are visible.
Is it visible now?
No, no. Now it is visible.
Is it visible? Yeah, okay.
Yes, start.
I'm Anam Zedi from the Department of Physics, BBD University, Lucknow, and the topic for my presentation today is structural, microstructural, sensing and optical properties of aluminum oxide and zinc oxide nanocomposite. Introduction. Nanocomposite materials have generated profound interest because of the new physics of nanoparticles and many potential applications based on improved electrical properties and superior mechanical properties like hardness, fracture strength, and fracture toughness. Nanocomposites may be defined as a distribution of a second phase of dimension in a matrix amorphous or crystalline line. Alumina is one of the most employed engineering ceramic, having great potentials in many special fields where low density, high stiffness, high hardness, chemical inertness, and good high temperature properties are required. Zinc oxide is a potential choice as a second phase because it has a high thermal, chemical, and mechanical stability in addition to its thermodynamic compatibility with aluminium. Till now, there are many researches on alumina-based ceramic nanocomposite, but a very little research attention has been paid to aluminum oxide, zinc oxide nanocomposite, and therefore this paper would be of great advantage for potential applications. Here you can see the XRD image of the nanocomposites. We can see that there are peaks of both the zinc oxide as well as aluminum oxide, showing that a nanocomposite has been formed. Along with it, we can see that there are certain impurities. We can see that a new compound has been formed, which needs to be removed from the nanocomposite by several methods. We can see the same image here. As we can see from the images, it is evident that as the percentage of aluminium is increasing, the density is decreasing, and the porosity of the nanocomposite is increasing. This is the SEM, that is our surface electron microscopy, which gives us a surface structure of the nanocomposite that has been formed. And the last one that we have conducted is transmission electrons microscopy that would tell us about the internal structure. And here we can see that there are dendrite-like structures, and with the increase in the percentage of alumina, the density is decreasing and the porosity is increasing. And with the increase in the porosity, what happens that there is better adsorption or the absorption of the particles, which results in better sensitivity of the particles. Thus, I conclude that the introduction of aluminum in zinc oxide increases the porosity of the nanocomposite, resulting in better adsorption or absorption, and therefore the sensitivity of the sample increases. Thank you.
Okay, ma'am. Now there's correct. I'm sharing it. Zoya, I'm reading.
Zoya, yeah.
Good afternoon to everyone. This is Zoya Amreen under the supervision of Dr. Meenakshi Singh. My presentation is based on educational impact due to Corona. We all know that due to Corona, the education system is disastrously affected. If we go with the current worldwide epidemic, we can say that education is the most affected field in terms of corona. Not only that, people who are living in the poor states or living in the old rural areas, they are not getting the good connection, because of which they are affected a lot. And in according to February 22, the lockdown was shut down. The people are all over there. There is a lot of connection problem. Everything we suffered, and that made the children suffer a lot. Then go with, I say that in every family, there's not necessary that every family have a Android mobile, or we can say they have a good connection. That affect a lot. If I go with the education report of the analysis, we can say that the parents are totally affected. They get devastated, not only because of the corona, but because of the education, because the children were too much involved in the mobiles, in the terms of the network, that they are affected. They're not going towards the education, but they're going to different other fields, because they're getting sort of connection. And as you know that in internet, it's the who wants to get an education. So what the people have tried, they have taken the traditional education terrain, like people like started those education system which is under the government there. They used to put a presentation in the form of the blackboard or somewhere. Now, how they deal with the education? They don't know how to deal with these network. They don't know how to deal with the Google Meet or the Zoom meetings. They are unaware of it. So first and foremost thing, that's the biggest the difficulty that they are suffering. Teachers, whether it's seminars or whether it's students, everyone is totally affected beyond it. Then it's taken expert, how the expert go. So what we done, that education system is affected in every field. They have lots of, like the children's were totally on the blackboard, the chalks, the books was being there, they come out, they have a good, and that makes them to come in the depression sector too. The children are suffering because they are making themselves helplessness. They are not unaware what to do, how to give the examination. They don't know how to do the examination centers. Moving on to it, we can say that seminars has been done up. Yes, there are good manner also, but I think in the corona cases, the education is the most affected field. That anything, if I go with education, I think that is the most devastated form. So coming with it, that the possibility is this, that there are parents who are allowing the situation. Not only that situation, they have given them mobile, they have given internet connection, they have given everything, but still the children don't want to study because they are getting some more thing in it. On the conclusion background, I just want to say that COVID has not only made us to understand that how to deal with the corona cases in the terms of education, we have become more advanced, we have become more about the connections, and I think this is the good one. Corona has made us good goal to be an education specialist. Thank you to everyone.
Okay, thank you.
Next, Dr. Casey Dubey. Dr. Dubey, are you here? Present is Harshal Naresh Rathore.
Which number?
Rathore.
Okay.
I think he's next. Chinky.
Ma'am, am I audible?
Okay, yeah. Yes, you are audible. Please continue.
Yes, ma'am. Ma'am, is my screen visible?
Yeah, it's visible. Continue.
Today, the topic for my presentation is energy resources and environment conservation. We all know that in 2022 or before 2022, energy rates have put a strain on Earth. So, to solve this problem, we must use renewable resources of energy, and solar energy is the most of energy.
So there are few applications of solar energy that I will be discussing in this presentation. There are three main ways to harness solar energy. First one is solar heating and cooling. Second one is concentrating solar power, and third one is photovoltaics. Solar heating and cooling. We all know what solar heating and cooling are. Solar heating and cooling basically uses the thermal energy from the sun and then heat water or space or air. These systems are used to heat pools or the residential area. The new topic that I want to discuss is concentrating solar power. Concentrating solar power plants uses mirror to concentrate the sun's energy to drive traditional steam turbines or engine that create electricity. The thermal energy concentrated in CSP plants can be stored and then used to produce electricity when it is needed, day or night. The next topic is photovoltaics. Photovoltaic devices can use to power anything from small electronics such as calculator, road signs, or homes. There are two creative uses of photovoltaics that we have seen. First one is solar power load, and second one is solar floating farms. The solar power loads have the ability to generate clean energy, illuminate themselves at night, melt snow and ice during the winter months. Factors affecting the energy production include panel efficiency, panel angle, tracking method, sunlight hours, and temperature. Next topic is floating solar farms, or also known as photovoltaics. These are the photovoltaic solar power system designed to float on the large surface of water bodies. Studies also show that this power production of a floating solar panel is greater by up to 10% due to the cooling effect of water. It also reduces the evaporation loss of water and suppresses algae blossom, lowering water treatment costs. Thank you for giving me an opportunity to present this seminar. Thank you very much.
Thank you so much.
Thank you so much for being in time. Next, Chinky Gangwar.
Yes, ma'am. Good evening, everyone. I am Chinky Gangwar. My topic of presentation is the synthesis of argentum nanoparticle by the chemical reduction method and its characterization. Nanomaterials are those materials whose one of its dimension lies in the range of 1 to 100 nanometer. Particularly, I have chosen noble metal nanomaterial because they have large surface area to volume ratio because of which they have high diffusion rate, and their feasibility at lower temperature. Also, they possess unique physicochemical properties because of which they can utilize in different fields such as electronics, imaging, biosensing, pharmaceuticals, etc. Now, why argentum nanoparticle? Because of its biological importance, mainly it shows highest antimicrobial activity among all the noble metals. That's why I have chosen AgNPs. And also the cost of silver salt is less as compared to other noble metals. Now, why chemical reduction method? Since the chemical reduction method is simple and there is ease in handling of apparatus, so I have chosen this method, and also the yield of a product is high. Now, in chemical reduction method, I have chosen tannic acid as a reducing agent because it is a plant-based extract, so it leads to a environmentally benign synthesis and cost effective for large scale synthesis, and also it leads to reduce the extra chemicals because it behaves as a 20 chelator molecule. Also, it shows highest antioxidant activity and anti-carcinogenic activity, anti-mutagenic activity. This is the structure of tannic acid. Now, the synthesis method involved in the formation of AGNPs. I have chosen 3-neck flask, which is fitted with a condenser to avoid the evaporation of the mixture. In this 3-neck flask, I have taken silver nitrate solution, which is thermally equilibrated at 30 degrees Celsius, and from the side arm, I have injected the mixture of tannic acid and NaOH, which is also thermally equilibrated at 30 degrees Celsius, and stirred this overall mixture for 15 to 20 minutes at 3000 RPM. Then there was a change in color was observed. The actual silver AGNPs I have synthesized in lab looks like that, and these nanoparticles were characterized by several techniques such as UV, FTIR, SEM, TEM, XRD. This is the absorption spectra recorded from UV visible spectroscopy, and this AGNP colloid shows a sharp SPR band at 410 nanometer. However, no such peak was obtained in the absorption spectra of a tannic acid. This is the FTIR spectra of AGNPs, and it shows several bands which confirms the formation of AGNPs, and it also proves that the AGNPs is stabilized by several groups. This is the FESM image of the AGNPs, and it shows that the AGNPs are agglomerated in the solid phase, and they have pseudo spherical shape. Now, for further confirmation of morphological analysis, I have performed TEM analysis, and from this it is clearly visible that they are spherical in shape, and the size of the nanoparticles are quite different. And from histographic analysis, it has been observed that most of the nanoparticles are size 15 to 20 nanometer in diameter, and the size distribution is non-uniform. Further, for lattice parameters, I have performed PXRD analysis, and there are 4 diffraction peaks corresponding to different HKL planes have been observed, with the most prominent peak at 111 HKL plane, which ma'am completely ho gaya, which shows that the average crystallized size is
There are among 20 amino acids in our daily life which are essential and as well non-essential, which may be consumed by the food products we eat, or they can be synthesized inside our body. Lipids like fatty acid, triglycerides, which include fatty acids, fats, phospholipids, steroids, are also necessary. Our basic human cell nucleus consists of nucleic acid, which is the genetic material in all the living beings. It has sugar, phosphate, backbone, white.
My topic is environment safety management. Introduction. To safeguard the life of human beings due to drastic change taking place in environment. Environment safety is necessary. Otherwise, life will not be safe on Earth. We will study how to monitor the changes in the environment and how to reduce their impact technically. Reduction method. Emission can be purified with filters. Gas from the process are fed into the scrubber were spilling with water and clean air went out from the chimney. Mostly these gases are chlorine, sulfur dioxide, nitrogen dioxide. Sometimes alkalines are added to splitting water to increase efficiency or scrubbing gas. Dissolved in water. 3 prime factor of environment safety management. The aim of environment safety management is to balance economic and environment safety. Management procedure, collection of information, decision making, management principles, principle of payment. It is based on economic assessment of natural resources, natural lane participants in the production process along with the production. Principle of scientific validity. It is required for the economic assessment of natural resource and the calculation of environmental damage. The principle of economic responsibility, the principle of comprehensive, the principle of economic calculation. Institutional method. It is based on law, order, instruction, and other regulatory documents. Economic method is based on material, interest, funds, and subsidiaries. Social psychology method. It is based on belief in environmental literacy of people, the traditional value of safety. Examples. Which plan to achieve zero carbon footprint in the production till 2030. Already they use recycled materials and clean energy sources. Total an oil and gas company has a program to reduce its impact on the environment. It includes a goal to achieve zero emission from energy production by 2050. And there 60% reduction in average carbon intensity of energy fuel worldwide. That is why companies are using renewable technology, occupational safety and health, environmental control, chemical safety produced by factories, monitoring of environmental impact, control of environmental impact source, monitoring of environmental risk. Conclusion, human race has adopted above methods sincerely to save the life of humanity. It has to be seen that global warming reduced and where it is possible the environmental factor. Thank you, ma'am.
Thank you.
Thank you.
Next is Dr. Rajendra Kumar. Dr. Rajendra
Yes, ma'am.
Kumar.
Yes, ma'am.
What is his number? They are not in the list.
Third number, ma'am.
Ma'am, he was at number 3. He was having some technical problem. That is why he was not audible.
Okay.
OP03.
Yes, ma'am. Good afternoon to all of you. My topic is aqueous chemistry of central component of B12. That means cyanocobalamin and essential element copper with cytidine triphosphate and nicotinic acid. Introduction. Here the derivative of nicotinic acid is pyridine carboxylic acid, like picolinic, nicotinic, and isonicotinic acid. Nicotinic acid is also known as niacin, is a water-soluble vitamin B3, which is used for the treatment of schizophrenia, hypercholesterolemia, diabetes, osteoarthritis, autoimmune disease, and pellagra. The overall stability constant is given by the equation here mentioned, and their terminology is mentioned here. L1 stands for the cytidine triphosphate and L2 stands for the nicotinic acid, and the stoichiometric numbers PQ, RS, T are either 0 or positive integer, and T is negative integer for a protonated species. Objectives: to determine the speciation and stability constant ternary and quaternary complexes in solution by potentiometric method, to determine the protonation constants of free ligands, and to obtain the titration value of ligand and its metal complexes, to analysis the titration data through Scotch computer program, which is a known stability constant of generalized species, and used for the determination of percent species formation. To plot speciation curve of obtained species. Here method, the ionic strength of all mixture solution was kept to 0.01 molar sodium nitrate. The free acid concentration was kept to 0.02 molar in each case. The following sets of solution were prepared, keeping the total volume 50 ml in each case. The molar ratio of binary, ternary, and quaternary system was taken in same ratio respectively. Here the composition of the solution taken in each. here आपको quaternary system copper cobalt CTP and Na system formation of copper cobalt means cytidine triphosphate and nicotinic acid what's shown in figure 1. Graph of this quaternary system reveals the following species: cytidine triphosphate, triproton and nicotinic acid monoprotonated, copper and cobalt monohydroxide binary complexes of CTP with copper and nicotinic acid with copper, and CTP with cobalt, CTP nicotinic acid with cobalt. Ternary system is copper CTPNA, cobalt CTPNA, and quaternary copper cobalt CTPNA. Here, a species formation curve of quaternary copper cobalt CTPNA system is shown. These species are formed by Scotch computer program. Here S3CTP protonated ligands and diprotonated monoprotonated, and their hydroxide are both metal and cobalt. Binary system of cobalt CTP, cobalt Na, and copper with both ligand, cobalt with both ligand, and copper cobalt with both ligand, means quaternary system. And these graphs shows the formation of concentration at 9 pH maximum reaches about 99 approximate result. The multi-metal multi-ligand complexes involving more than one metal ions and ligands. It is capable of calculating simultaneously or individually association constant for any of the species. SciC developed a new computer program, stability constant of generalized species, and conclusion. Stability constant and other related constant of binary and ternary complexes of cytidine triphosphate and nicotinic acid with different metal ions in aqueous solution at 37 plus minus 1 degree Celsius and having ionic strength 0.1 molar sodium nitrate. Proton ligand formation constant log beta OPQRS of the ligand, that means the dissociation constant, and here given the value of protonated species of both ligand cytidine triphosphate and nicotinic acid. The hydrolysis constant.
Kindly conclude, sir.
Rajan, Kumarji, please conclude your.
Yes, ma'am. Yes, ma'am.
And the last presentation is Krishna Yadav.
Okay, thank you. Thank you.
Thank you. Thank you, ma'am.
All the participants now. The last is Krishna Yadav.
Okay, thank you, ma'am. Just sharing the screen. Wait.
Number 29.
Ma'am, 29. 29. Yes, ma'am. 29.
Okay.
Ma'am, my screen is sharing.
Not yet.
Not yet. Achha. Now, ma'am?
Yeah, it's visible. Kindly continue.
Okay. Thank you, ma'am. Namaste to everyone, whosoever is present here. My name is Krishna Yadav, and I am Associate Professor from the Department of Mathematics, Mahila Degree College. And topic given to us, health, hygiene, and environment. I've chosen the health part. And health, of course, it consists the three type of the health: physical health, social health, or the mental health. So if mental health is there, so we can cover the social as well as socially healthy, or as well as the physically healthy. So for mental health, socially, I have taken the spirituality part, and for that spirituality part, I have taken some mathematical concept in that. So that mathematical concept, I gave the findings because time is very less. So how the numbers is giving the help to understand the meaning of the certain mantras, and that mantras gives us the focus. And when it is focused mantras, that means it gives the immunity to mental immunity, like a physical immunity, the kadha and all these things in COVID. So mental immunity, by reciting the mantra, we can be stronger in that case. The two numbers are very prompt, that are the 0 and infinity. These 0 and and infinity, I have taken one by one. That is the Vedic platform, that is the Vedic philosophy, then the scientific approach, then its application, and then the findings. So Vedic philosophy, it says that कि यदा सर्वे प्रमुच्यते कामा ये शस्त्र हृदय स्थिता आत्मृत्यो मितो भवयत्र ब्रह्म समश्नुस्ते. So that means कि that is when all the desires in a heart of a human being is reduced, and that are represented in the mathematical modeling, that is the limit of a d tending to 0, p by d. What is the p? P is the position, d is the desire. So denominator is reducing. The value of a denominator reduces, the total value gives the more and more. So that means if it is a 0, so we'll get the infinite. And what is that infinite? That is the Paramatma. In that form, I wanted to taken up this part. Second part of this, that is the second part of which is taken at the Brahma's put part. Ado radyan mahanto mahyam natmam guhyam vihato yasya janto. That means that is smaller than the atom and greater than the greatest. The soul resides in the heart of the living being, and this is known. That is the how immortality soul is there. So, and same way, I have taken the scientific approach also. So that scientific approach, that means ki how this that is our solar system. This I can say the scientific approach, that is the Milky. What is the galaxy? That is the Milky Way. And galaxy is what the set of the solar system. Then that solar system, of course, the sun, planets, satellites, stars, all things like that. To cover the one galaxy takes the 2 lakhs light year. And one light year is 3 into 10, speed of a light is 3 into 10 to the power 8. So to cover one end from another is 250 billion years. So how it is impossible to find the size of that. And the universe, now the concept of the universe has also been changing. New theory says that instead of a universe, we have got the multiverse. तो कितना समय लगा होगा एक एंड से दूसरे एंड में एक यूनिवर्स को जाने में? 250 billions once it is taking. So it is a multiverse. So the Almighty who made this this universe or the multiverse, so it is not possible to find the shape and size of that. Only it can be realized. The concept of multiverse and inverse, that is very interesting. That is the finding the Paramatma. Paramatma, that is the shape, that is the formless. And scientifically, I can say the bundle of energies. So these things, of course, cannot be visualized by the naked eye. But how we can do it? By concentrating ourselves. Everything is present in our body. So that mantra I have taken is, that mantra Isha Vrihadaranya Upanishad se jo liya gaya hai, ki Om Purnamada Purnamidam Purnat Purnamudaya Purnasya Purnamadaya Purnameva Vashishyate. So Om, that is the unstruck sound. That usually we are getting the sound by instructing, but if this sound is unstruck, keep on going on in this universe. And Purnam, that means the complete, that is 0 and infinity. So in mathematics, determinant forms are there, 0 by 0, infinity by infinity. So how to define these things? So here it says Purna, complete. Ada, that means the Ishwar. Purna Idam, that means the words given by the God, that is also complete. And once we are taking the something from the Purna, Purna में से हम पूर्ण निकाल रहे हैं, तब भी वो पूर्ण ही बच रहा है. और पूर्ण में पूर्ण डाल रहे हैं, तब भी पूर्ण ही बच रहा है. So what is that? That is the Paramatma. Then how can we define? So that is the symbolically we are taking, that is in the form of a Shivling. Shiv is the form of that Paramatma. And when we are taking the ling, Shivling, upper side of that part, it is the half ellipsoidal. And our Earth is also known as the irregular shaped ellipsoidal. So my epilogue, that is the conclusion of the chapter, is that the concept of the infinity and the zero number system lie at the root of the mathematical analysis, which is growing like a banyan tree in the various direction. And this implies that the above basis mathematical concept, which originated in India during the Vedic periods, provided a solid foundation for the development of the mathematical analysis and its application. And second findings, what I have got, it has an indeterminate form in mathematics, that is 0 by 0 and infinity by infinity. Many more are there, not only 2. Doesn't mean that it is meaningless. The value of determinant can be evaluated by using the certain transformation. And that transformation, that is the process. So, of course, the supreme of the supreme, which is formless but can be realized. And in this process of doing this, that means, of course, immunity will increase. Which type of immunity? Mental immunity will increase. So when mental immunity increases, so of course your physical and social immunity will increase. And that's what my concept, how to fight the not only in COVID, in the day-to-day life problems. Thank you, ma'am.
Okay, thank you so much, Krishna.
Thank you, ma'am.
Now we have finished with all the presentations. May I request respected Dr. Sushma Trivedi to kindly take the session again from here?
Yeah. Thank you very much, Dr. Neha Agrawal. First of all, I would like to thank Dr. Rashmi Srivastava for her patience. She has been here for more than 2 hours and very patiently she has been adjudging the presentations and encouraging the presenters, because they are all young presenters. They have put in lots of hard work in their presentations. So you have been a very supportive kind of chairperson. Thank you very much, Rashmi.
Thank you so much.
So now we are in the concluding part of this international conference. We have been able to initiate a discourse, a dialogue on how to engage chemistry more and more for human well-being. Our team of organizers has done really well. We had very informative talks and we have very lively discussions and question answer rounds. So now it's we are in the end of the conference. To host the valedictory session, I would like to invite Dr. Shobha Mishra, Head of the Department, History of Navyuga Kanya Mahavidyalaya. So I would request Shobha ji to take over the mic.
Thank you so much, Sushma. I hope I am audible, Neha.
Yes, you
Can you hear me?
Yeah.
Okay, well, first of all, very, very everyone. Sushma just told you. Professor and Head Department of History. It is not from my side that somebody's phone was ringing on the other end.
Please mute yourself. Dr. Renu Agarwal.
कर लिया उन्होंने म्यूट कर लिया
है तो मैं कंटिन्यू करती हूँ फिर
Yes, ma'am.
So I take this opportunity to welcome each one of you to this valedictory session of the 2-day international conference on chemistry for health, hygiene and environment. The conference, as we know, was organized in collaboration with the Chemical Research Institute of India, which was established in 1999 and is dedicated for organizing seminars, conferences, workshops in all branches of chemistry, and also in association with the Association of Chemistry Teachers, which was launched in 2000. And the purpose was to serve as an apex national body of chemistry educators. We know that the conference witnessed varied dimensions through its course of 2 days, 4 highly beneficial sessions, and of course, a lot of presentation. I say beneficial because as a scholar of history, I hardly understand chemistry. But in the past 2 days, I have realized how chemistry, along with a few healthy practices, is conducive to maintaining health and preventing disease. I have understood how clean air, stable climate, adequate water, sanitation and hygiene, safe use of chemicals, and sound agricultural practices are a prerequisite for good health and good environment. Chemistry is there in each and every aspect of our daily life. It is in the food we eat, the water we drink, in the tears that fall from our eyes, the cosmetics we use, and even in the baking powder we use to make our cakes more fluffy. In fact, the practical aspects I have are far more widespread than its aspects in the laboratories, perhaps. In fact, chemistry, when properly used and understood, significantly contributes to the improvement of our life quality. On the other hand, if misused, it can be highly hazardous and negative for our health, for our life, and of course, for our very existence. A very recent example of which is the war situation that the world has been gripped in because of the Russia-Ukraine conflict. The bombs, the missiles, and the mushroom-shaped clouds that appear as soon as a heavy-duty bomb touches the ground are highly devastating to our civilization. Can we live in such an environment? This is not what is needed of chemistry. And I'm happy that fortunately in our 2 days conference, we have been talking about the positive uses of chemistry in the field of medicine, in the field of health, in the field of hygiene, and in the field of environment. A very happy, peaceful, serene, and conducive environment. To continue with this valedictory session, I now invite Dr. Rashmi Srivastava, our guest for today, and of course, a judge for the presentation sessions, for a kind words.
Thank you, Shobha ji. Thank you so much. And it is my pleasure to participate in the valedictory function of Multidisciplinary International Conference on Chemistry of Health, Hygiene and Environment, being organized by one of the best college of our city, Navyuga Kanya Mahavidyalaya. And I congratulate the principal of the college, who is renowned educationist, Professor Manjula Upadhyay. It is really a proud moment for the college to organize an international conference under the auspices of Association of Chemistry Department and Chemical Research Society of India. I know both the organizations are doing excellent work in the field of chemistry and its applied aspect, and regularly organizing conferences, workshops, and seminars, etc. Heartiest congratulations and best wishes to both the organizers. And I'm happy to note that during the 2 days deliberation, eminent scientists and scholars presented their findings and interacted with the participants. Today I heard the lecture of Dr. Upreti. It was really the nice one. And it is heartening to note that lecture was organized under the number of themes vital in the present day scenario, like biological, medical, and pharmaceutical chemistry, water resources management and water waste management, health and hygiene, energy resources management, and whatnot. And it is very interesting that mathematics for everyday life has also been chosen and discussed in the international conference. I am happy to see the name of international scholars and scientists of different disciplines, especially Professor Dr. Upreti, Dr. Anant Pandey, Professor Satyanarayan, Dr. Sanjay Kumar Pandey, Dr. Gola, and Dr. Kanchan, etc. I have also come to know that Navyuga College scholars and MIT scholars have also presented their papers. They deserve congratulations and good luck for their future endeavors. I'm thankful to the principal of college, Dr. Manjula ji, and organizing partners, Association of Chemistry Teachers and Chemical Research Society of India, for inviting me to participate in the valedictory function. And the conference was excellently organized international conference representing dignitaries from different countries as well as scholars from our own country. Thank you. Thank you very much, and I wish a good luck and best wishes to Navyuga Mahavidyalaya and Association of Chemistry Teachers and Chemical Research Society of India. Thanks to Dr. Neha Agarwal for coordinating with me and inviting me to chair the session also. Now, I will announce the best name of the best participant when Neha ji will provide me the name of that participant. Thank you so much, Shobhaji. Thank you so much.
Thank you, Dr. Rashmi Srivastav, and a personal namaste to you. And actually, kudos for having we are very happy to have a very patient chairperson with us. You have been, just as Sushma said, you have been very nice, very cooperative, and very supportive to our presenters. It was, I actually thank you for your very presence with us today.
Yes, it was my pleasure. Thank you so much.
Now, I think as we are heading towards the closure of our today's event, I would now request Neha Agarwal, Dr. Neha Agarwal, to please come up and read her report as well as declare the results.
Thank you, Shobha ma'am. Thank you so much. I would like to give a small report of the two-day conference. We were having day 1 and day 2. Total, we are having 4 sessions. So the day 1 of the conference began with the inaugural ceremony, including a welcome note by Professor Manjula Upadhyay, the principal, Navyuga Mahavidyalaya. Then we had the first technical session that started with the welcome note of the keynote speaker, Professor Satyanarayan, Professor in Anatomy and Cell Biology, University of Florida, by Dr. Sangeeta Potwar. Professor Narayan discussed thoroughly about the novel therapeutic development for colorectal cancer. He said that cancer is a function of predisposition, exposure, and age, and told that colorectal cancer is the 3rd major health problem worldwide, and advised NSC49L treatment to it as a future second-line therapy. Dr. Sonal Agrawal introduced the second eminent speaker, Dr. Alok Ranjan, who is currently a research scholar in chemistry, Stanford University, California. He enlightened us on progress towards HIV/AIDS eradication, and he depicted various charts and graphs showing kit and kill approach to diminish HIV. The second technical session began with the introduction of keynote speaker, Mr. Anand Pandey, the President and Chief of Operations, Jubilant Ingrevia Limited. And the introduction was given by Dr. Pratima Ghosh. He shared his views on environment and sustainability, and he talked about the conservation of finite resources available in our environment. The second eminent speaker was Dr. Sanjeev Kumar Ocha, whose formal introduction was given by Dr. Seema Sarkar. He talked about the 5 elements of life and discussed thoroughly about medicinal plants and fruits like tulsi, neem, aloe vera, amla, and a very important thing, the pomato plant, which was a combination of potato and tomato. He concluded his talk by introducing the best 6 doctors. They are sunshine, water, rest, air, exercise, and diet. In the session, we had oral and poster presentations where Dr. Seema Joshi, Head Department of Chemistry, IT College, was the chairperson. And the vote of thanks was given by convener Dr. Neha Agarwal. Then we had day 2, which began with the Vandana of Ma Saraswati, which was performed by Ms. Tanu Kashyap, the student of Navyuga Mahavidyalaya. Followed by 3rd technical session. The keynote speaker was Dr. Sanjay Kumar Pandey, Chief Scientific Officer, Triangulum Biopharma, San Diego, USA, whose formal introduction was given by Dr. Anurima Banerjee. He discussed about RNA-based therapies as an emerging platform. He explained about several categories of RNA targeting, small interference RNA, microRNA, messenger RNA, and aptamers. He further said that there are 8 ASO-based drugs and 3 small interference RNA-based drugs that have been approved by FDA, and many drugs are there in clinical development. The second eminent speaker was Dr. Upendra Rao Gola, whose formal introduction was given by Dr. Sunita Singh, and he discussed about the emergence of green nanotechnologies for sustainable environment in detail. The technical session was concluded with the vote of thanks by Dr. Sunita Singh. The fourth and the last session was organized by Dr. Sushma Trivedi, where Dr. D.K. Upreti, FNA CSIR Emeritus Scientist, was a keynote speaker. He opened up a spectacular world of lichens, an organism which has the potential to produce natural substances which can contribute in the development as a safe drug. He informed about potential of lichens as metabolite for producing nanomaterials. He also talked about the ancient medicinal texts like Charak Samhita, Sushrut Samhita, which reflect our knowledge of lichens and their medicinal properties. He emphasized the need of modern research in untapped ethnic medicines, which are great source of knowledge of lichens. Then we had posters and oral presentations where Dr. Rashmi Srivastava was the chairperson and the chief guest of the session now. From here, now we will proceed for the valedictory session, and which is being organized by respected Dr. Shobha Mishra. Thank you, ma'am.
Thank you, Neha. After Dr. Neha Agarwal's detailed review of the 2 days event, we now move forward to that part of our event when it is time to feel relieved and also time to say thank you to each and every person who has contributed in making this a successful event. As far as the results are concerned, I think Dr. Neha will declare it after some time.
Okay. Ma'am, I hope that actually the results have been prepared after the 2 days judgment of Dr. Seema Joshi and Dr. Rashmi Srivastava. So yes. We will calculate the total marks, and we have calculated in fact, and the results have been decided on the basis of all the criteria, the total time taken and the matter, subject matter, and the way of presentation and the content which was developed by the participants. So the results have been sent to the respective Dr. Rashmi Srivastava. So I would request ma'am to declare the result. That would be more comfortable. I'll feel more comfortable.
Okay, very nice, very nice. Dr. Rashmi Srivastava, will you please do the honors?
Okay. The best presentation is Sukriti Singh, and best oral presentation by Dr. Kumar Swami Gellapalli. Thank you so much, Nehaji, for providing me. Okay, thank you.
I should take over now. I once again welcome each one of you to this warm and beautiful evening. It's my pleasure to propose a vote of thanks at the valedictory session of the 2 days international webinar on chemistry for health, hygiene, and environment. On behalf of the entire fraternity of our institution, I extend my prayers and thanks to Lord Shiva, who continued to be at our side as we sat on board on the auspicious occasion of Mahashivratri. I extend a hearty vote of thanks to all our expert speakers, Professor Satyanarayan, Dr. Alok Ranjan, Dr. Anant Pandey, Dr. Sanjeev Kumar Ojha, Dr. Sanjay Kumar Pandey, Dr. Upendra Rao Gola, Dr. Ranjana Kanchan, and of course, Dr. D.K. Upreti. I thank them because despite their very, very busy schedule and our untimely hours, they were kind enough to listen to our request and to grace our event. We had an opportunity to listen to their enlightening speeches and actually benefited from them. We have gathered a lot of knowledge in the past 2 days, which will truly help us in our life. We are very grateful to your valuable presence with us. And a special vote of thanks to Dr. Seema Joshi and Dr. Rashmi Shrivastava for sparing a large part of their valuable time with us and being very, very, very patient. We all know that an event like this is a result of continuous and arduous efforts of an entire team, which actually turns the wheel rolling weeks before the event actually happens. It requires a lot of planning, coordination, and a highly vigilant and watchful eye. We have a special mention and gratitude for our principal, Professor Manjula Upadhyay, for always stimulating us to do our best. She deserves special commendations for her untiring efforts and constant support, for always being there as a guiding force. It was also my pleasure to acknowledge the contribution of each and every person of the chemistry department who worked really hard to make this event possible. A special thank you goes to Mr. Sunil Verma for handling the technical support system, very essential for the flow of the event. The team's days of hard work was visible to us all in the past 2 days. I'm also gratified to all our audience, all our presentations, and our time with us. It has been a great pleasure. I wish you all a very, very great day ahead. And इस ख्याल के साथ कि मुसाफिर है हम भी, मुसाफिर हो तुम भी, किसी मोड़ पर फिर मुलाकात होगी. I am Dr. Shobha Mishra on behalf of Navyuga family signing off for our 2 days event. Thank you so much.
Hello, ma'am. Hello.
Hello.
Ma'am, my abstract is not there in the book which you have sent in the WhatsApp group.
That we'll discuss, Bushra ji, we'll discuss it later on. Actually, you sent it very late, that is why.
It is a request that if you have the session has been closed, the event is closed. If anybody has any personal problems, they can kindly contact Dr. Neha Agarwal or Dr. Sunita Singh later.
Okay. Okay, ma'am. Thank you.
Thank you so much.